Just a note on the facilities that are represented here and not represented here and how that came about. Obviously, you all know there are other facilities not represented here that are doing amazing work. We have friends and esteemed colleagues in Orangeburg, Tennessee, Neosho, Genoa, all these places that we know that are doing great. This is not an exclusive or exhaustive list on who's doing good work. Um, we just thought, as we were talking about how this might work, that we might be able to fit in 9 hatcheries. That seemed doable to go through in a day at 30 minutes each. And the idea arose here at NC State as we were discussing the possibility of having some sort of regional meeting. So you all represent labs that are close to us relatively geographically and people that I know that do good work. So if there's anyone that's hears about this in the future or watches this in the future from outside of this group, you can be mad at me, but please don't. And I would say let's just arrange a similar meeting with another group of 9 in the future.
So I'm here today. I have a couple partners with me. I think maybe at the all the way end of your list, Zach Taylor is now with Virginia DWR, but also with us today is Dorothea Miltenberger, Megan Kabbalah, and maybe Sally Ellers. So I'm going to give just a high-level overview of mussel propagation and some restoration projects ongoing in Maryland. I'm now currently the administrator of the Freshwater Mussel Assessment Program. And although my part of DNR is separate from the Hatcheries program, I work pretty closely with them in an advisory and collaborative role. So the main driver for our mussel propagation is something we call the Freshwater Mussel Restoration Initiative. It was born out of a FERC water quality settlement agreement. And if you notice by the map, the majority of that restoration work actually does and will not take place within Maryland. It's due to a large hydroelectric dam blocking mussel host migration and access to almost the entirety of the Susquehanna River watershed, but also some direct impacts from the project's existence and its operations downstream. This was a pretty unique FERC settlement in that the operator was requesting a 50-year license. As such, the agreement is supposed to provide 50 years of financial support. So this is probably going to end up being not only one of the spatially largest restoration programs or projects for mussels, but also the longest temporally. So it provides money for restoration and propagation planning, hatchery design and construction and operations, a mobile mussel propagation trailer because things like design and construction take a long time, and the people that negotiated this settlement agreement didn't want to patiently wait for bricks and mortar. And then some alternative actions like mussel translocation, host stocking, kind of pairing other on-the-ground restoration activities that were funded through other parts of the settlement agreement to benefit mussels. The license is currently in mediation and is supposed to be reissued for essentially a new 50-year calendar within the next few months. We're currently in the 50% proceeding to 65% hatchery design and process, where it'll be a large industrial style facility located in Brandywine, Maryland. And this is because there is an existing fish hatchery within department-owned lands. So on the right, you can kind of faintly see some existing sketch lines and ponds where a large fish hatchery already exists. you can see kind of just some high-level sketches of what the grounds of this mussel facility would look like, with new ponds excavated to culture mussels and provide source water to the building, new ponds for fish host culture. And then the lower drawing is our current kind of 50% sketch of the facility, showing kind of our offices, our effluent and influent treatment rooms, meeting space, and then fish and mussel spaces. Because this settlement is largely driven by kind of mussel community impacts that are more ecosystem driven and not rare species driven, we kind of have a little different operational paradigm where we're looking to produce large numbers of mussels in the least costly way, and not necessarily rare species unless the situation dictates it. The design began in 2024, and really in the next week or two, it should hit the 65% design phase. There's currently a $6.5 million estimate for construction, and construction is projected to go through at least 2026. That said, we are conducting some mussel propagation within the fish hatchery. The hatchery operates seasonally, does not have a lot of recirculating operations within it. It's a pretty extensively operated hatchery. So within that, we have capacity to isolate mussel broodstock and host fish. We have at least 5 multi-tank recirculating systems for isolation and infestation. And then we have just a recently kind of revamped juvenile culture system set up, which includes things like mucket buckets, up and down welling buckets within raceways or sumps, various sediment culture boxes, with a capacity for about 150,000 juveniles at drop-off. Although the reality is we're working with numbers that are 1 to 2 decimals less than that. Again, because we're at the fish hatchery, we also still have ponds available for grow at any time. This just kind of gives you an idea of what we're currently working with, including the lower left is a system that Zach applied from Chris's new facility. But some things like the upper left were just existing fish hatchery infrastructure that was not being used, that we replumbed and made useful for infestations. Our mobile mussel propagation trailer has been in operation for 2 years now. So this was money we carved out of the Freshwater Mussel Restoration Initiative. It's a 48-foot long race car hauling trailer with some kind of customizations. And in it, we can transform relatively easily about 25,000 mussels kind of per operation. So it runs for about 8 to 10 weeks in the spring and the fall at Susquehanna State Park, which is near the dam operations. And we have the ability to literally park it next to the existing fish hatchery and plug it in and operate it in the summer. And we're actually doing so because it provides capacity and also a source of grant match. So far, you can kind of see what we've done is we've kind of learned these applications of strapping things like refrigerators and AHABs and raceways to the floor of a trailer and operate within them. But we've averaged about that 25,000 mussel transformation goal. We also have this because it's forward facing. The mussel hatchery is located about an hour and a half away from the dam project within a state forest that is not publicly accessible. As you can kind of see in our right side pictures, this trailer is meant to literally open up to the public. It's ADA accessible. The sides facing the parking lot open up. We have a television with Bluetooth capabilities, so we can stick a camera in a fish tank, hook it up to our microscope, and people can either walk up to what we're seeing or see it visually from afar. we've averaged around 75 visitors kind of per 8-week season. And that's really without doing any outreach. This is just kind of passively letting people know that visit the state park. And then in the spring of 2024, we had a couple school groups and an environmental journalist conference happened to be in the area and came and stopped by. So you can kind of see some of the things we're doing in the trailer. So far, it's mostly been used for lamp saline production, but we've also used it for some anodontine production. One of our other large funding sources currently is a NIFWIF grant called program called Chesapeake Wild. So we have a collaborative project with partners in Pennsylvania, including Fish and Boat Commission, Dan Spooner at Lock Haven University. We're also working with Jess Jones at Virginia Tech to assist some of their needs. So this is focused on yellow lamp mussel and eastern lamp mussel. This project was identified in part because it's a river in recovery from a habitat and water quality perspective, but there are 2 low-head dams at the bottom of our project area that do not allow recolonization of mussels upstream. So we're going through a series of steps, including restoration planning, field work, a silo study, an adult kind of site fidelity study to kind of inform the propagation and stocking and monitoring. And then also integrate some education outreach because this is happening within the community's university in Lock Haven. So the propagation goals are largely set again around eastern and yellow lamp mussel. We're also looking at streamside infestation of eastern elliptio, and this is in part because of those alternative activities that DNR is required to do. The license for Conowingo Dam requires an annual trap and transport of American eels, which in the mid-Atlantic are probably the primary host for eastern elliptios. So this trap and transport already happens irregardless of mussels. So we have the ability to essentially make a request that eels go to a certain location to benefit mussel population dynamics. And this was actually shown to be successful in a prior study that USGS and US Fish and Wildlife Service did, where targeted stocking of American eels over existing eastern elliptio beds caused recruitment to be evident within just a couple years. So this is kind of in a developmental process and will proceed next year into implementation, where we have to kind of align the stars of eastern elliptio gravidity, American eel migration and transport with extracting an infestation. We've recently also received a kind of a one-time influx of state funds that we use to backfill hatchery design. We also use it to establish an in vitro contract with Harrison Lake to supplement kind of our operations, and then used it again for grant match. Through another NIFWIF program, we're conducting a large-scale restoration planning project for the Upper Potomac River. It's a very similar situation to the West Branch of the Susquehanna River, where we have a river that was impacted by kind of industrialization, logging. Conditions have by and large recovered, but again, a low-head dam is precluding natural recolonization. So this year we floated about 85 river kilometers to map and identify suitable habitat. We piloted a couple different survey methods that we will employ broadly this year throughout the 100 river kilometers. We've done some quadrat surveying and reference beds to develop kind of a restoration baseline. and use that to estimate our stocking potentials. We'll conduct most of that field work in 2025, along with a silo study using juvenile mussels from Harrison Lake to kind of hone in on potentially finer spatial limiting factors to mussel restoration. And then we'll do a little more field work in 2026 and kind of wrap up this grand restoration plan that will probably, by most estimates, call for another multi-decade approach, because there's probably in the neighborhood of 4 to 5 million mussels that could be supported in this habitat. We work locally with the Potomac River Keeper. This is a nonprofit water quality advocacy agency. So they're interested in mussels from kind of the ecosystem services perspective. This has been ongoing for a handful of years now, where we've done some in situ propagation and culture. This partnership has more broadly expanded because of overlapping projects with Virginia Tech and Virginia in the Potomac River, because that is an interjurisdictional river. And we also have some private support from the National Harbor Commercial Development and the marina operator. Unfortunately, in situ propagation that might work well in lakes and ponds, I can tell you unequivocally does not work well in tidal freshwater, especially in an environment like a marina. But in situ culture has been phenomenal. In species like eastern pond mussel, we have mussels that were about 9 months old and on average 5 millimeters grow to over 50 millimeters in 3 months once they were put into floating baskets in the Potomac. So it averages about 8 millimeters of growth a day. We've seen that with other species like eastern lamp mussel and alewife floater. And then our other ongoing project is a state wildlife grant project. It parallels the work that's been done in the brook floater comp swigs. Maryland was not a funded partner to that, but we used our own stateside money to do work in kind of parallel. This was the state's first restoration project with a listed mussel species. Brook floater is listed in endangered. The overarching goal was to restore species diversity and mussel community function. Again, this is a largely forested area that is recovered, but is isolated in this upper Potomac area from kind of the best remaining populations. So water quality, habitat, fish hosts, they're all available, but there aren't enough mussels to support natural recruitment. So since 2019, we have stocked 4 plots, pit tagged every mussel, and done monitoring in a couple different ways. At first, we looked at quarterly pit tag recoveries with submersible readers until the remnants of Hurricane Ida literally washed every mussel that was there and every mussel that we stocked away. Since then, we've maintained just an annual survey to measure growth and mussel bed density. With brook floaters, to be kind of overly precautious, we started with 2 years of cage studies to track growth and survival, just to make sure there was no kind of unaccounted for limiting factor. And in this case, our growth was in short periods was 0.5 millimeter a month or more. Survival was 90% or more. So again, over these 5 years, we've stocked just under 3,000 mussels of 3 species. And through the NIFWIF project for the Upper Potomac River, we're going to leverage those funds to expand this from 4 sites to 6 or 8 sites and kind of connect the dots from at least the Pennsylvania-Maryland border down to the Potomac River, and then also look into stocking mussels in Pennsylvania. And with that, I'm happy to take any questions. And if you have any questions that you think of, please feel free to direct them to myself or Dorothea, who is currently staffed at the hatchery.
Any questions, anyone? I know I've got a couple. Um, let's see. Yeah, let's see. So, Matt, I did have a question. I was wondering if you could talk about how y'all did your stream side infestations with the Elliptios. Like, how do you time that up? How do you deal with broods that are immature in a short-term brooder? And how does that work with big batches of eels that you're dealing with?
Zach may be the better person to answer this, but really this year was just more about um, taking kind of literature, past experiences with elliptio propagation in the lab, and past experiences with trap and transport, and trying to fit the pieces together in like a pilot fashion. And in reality, it was hard to do this spring because we had a cold spring. So Elliptioglockidia at the time, maybe the first eel transport happened, right? You couldn't get those two things to line up. Um, so in the past, what we've done with Eastern Elliptio is just bring them into chilled isolation units, and you kind of keep going out there until you get the right batch as close to possible. And in my experience, usually the first 2 or 3 trips, you're getting fertilized eggs and immature Glaucidia because you're really worried about being too late. And maybe on the third try, you get them a couple days before they start to release, and then you've got to figure out how to get the Glaucidia out of the mucus, and from there it's really just a matter of um, timing them up um, with the eel. The eel trap and transport happens daily. So for us, that's just a matter of saying we've got Glaucidia ready because these mussels are starting to extrude or the Glaucidia look viable, and getting that driver to then take a cut essentially. So there are, in any given time, there are tens of thousands of eels being transported daily. They're not allowed to hold more than a certain number at a time. So that's not the limiting factor for us. It's all about timing it up and having them meet us. So we have some things to work out still on the holding and timing side. And this year was actually kind of before our active period of the grant. So we used it more just to troubleshoot and apply these things we've done in the lab in the past to the field, maybe not successfully yet.
Nice.
And
And
I can,
no, go
oh,
ahead.
sorry, I can chime in a little bit on this. I was pretty involved in that project, and Matt did a good job. There was this past year was a lot of trial and error as far as timing is concerned, and this coming year, the plan currently is to try a couple different methods of collecting mature Glaucidia from Elliptios and lining up with the timing of the transports, which starts in early May, getting they get about 700 eels a day from the dam collection. And there were two or three specific things that we wanted to try. We wanted to run a small trial collecting Elliptios prior to spawning, then warming them up and spawning them in captivity, because last year we had problems collecting mature Glaucidia during actual spawning period. We never got to attempt to put Glaucidia onto eels due to river levels and other timing events that we just couldn't meet up properly. But we're looking to we were looking to try a number of different things to try and nail down a kind of best practice to collect the mature Glaucidia, and that was going to be the focus this coming year. But otherwise, I think Matt pretty much nailed it.
Awesome. Loretta.
Hello. Okay. Thank you, Matt and Zach. I had a question. Oh, I love the mobile trailer that you guys are doing. I think that was super cool. And I'm not sure if I missed it when you were talking about it. Are you planning to keep it or use it as an outreach unit, or what was your plan after your hatchery is built for it?
So I assume we will be required, right? So DNR receives this money from our State Department of Environment. So we come up with kind of a collaborative agreement. So they have some say in what we do with that money. I suspect we will maintain its use maybe in a more reduced capacity. We've talked about shifting the fall to like a mussel quarantining. Basically, we'll take the mussels out of the hatchery, we'll bring them close to the river, we'll start to temper them on Mussel River as part of a quarantining and tagging process. Because our propagation, while we can transform mussels that are gravid kind of early in the fall, they're typically not very viable. So we probably will be required to still operate it, at least in the spring. What we do with it kind of outside of that is really kind of on our own. But at least from the hatchery design perspective, it will probably park in a garage. So it runs off of just an RV 50 amp plug. So really any like campsite we can pull it up to and plug it in. It was in part designed to mimic, a lot of states have agriculture educational trailers that travel to schools. So really our limiting factor on mobility is we have a DNR has a semi truck that is used to haul this because it also hauls sturgeon and shad broodstock. So the availability of that CDL licensed driver is really what stops us. And then time, right? It takes us a day, almost a day to get it ready to transport, a day to launch it. So you're looking at like a 3 to 4 day operation just to get it somewhere to use it. So that's why, at least for the short term, I would say like the next 5-year period, it will largely kind of stay operating in the spring and maybe summer at the hatchery. So for the hatchery, for us, it's great. We plug it in, right? It's got air conditioning, it's got a Wi-Fi hotspot, it's got a fridge, whatever. It's essentially a lab on wheels.
Here, so it's solid people time and money and everything for an opportunity to reach folks like that. And I'm glad people saw by when you had it. So
Yeah, kids really like it. Right, it's sided with white aluminum, so it's literally a canvas. We can draw on it, we can stick stuff to it. It's a really good kind of immersive platform.
super neat. Thank you.
Well, I'm Andrew Phipps. I'm here at White Sulphur Springs National Fish Hatchery in White Sulphur. Most of you know me. I've been here since 2019, and of course, John Moore is here working with us. He does a lot of the system building and the mussel propagation, and certainly I try to assist and also stay out of the way. But we're not doing any kind of rocket science here, but we're happy to share what we're doing, and we're really pleased to be a part of this group, and hopefully, what we're doing will be helpful to some of you all, and we're looking forward to learning from you all as well. So a little bit about the facility. First of all, we're located here in White Sulphur Springs. We're at the fish hatchery. The fish hatchery's been here since 1900, I think. And we've been doing mussel work since around 1995. And we've got some really nice, interesting facilities here that are relatively biosecure, and we're really proud of them. And so we'll talk about those a little bit first. I think the most important thing that we have found when talking about facilities and mussel propagation is our water sources. And so here we run majoritively off of well water. We have a well that produces water at about 50 gallons per minute. We do use some of our spring water from the fish hatchery, but mainly we're using well water. The alkalinity of that water is about 56.2, which can be a problem at times. And the hardness is around 110. We run that well water directly into our systems, but we also run it into 3 1/4 acre lined ponds. You can kind of see the ponds there in the photograph. We then pull that water in. A couple years ago, we put in a pretty extensive 4-inch line system to where we could pull the water in at about 40 gallons per minute into all 3 of the buildings that are close to the pond there. And we're able to recirc that water, and we're able to also hold it statically inside in our systems. And so uh, we really rely on the pond water for food. We are filtering that water down to about 5 microns for most of our juvenile uh, animals, and we're putting those into the systems filtered to 5 microns and warmed up during the winter, cooled down a little bit during the summer. We use a whole pond water at times for our broodstock animals inside. We also use baskets, as you'll see, and so we're using that whole pond water for outside culture. For supplementing, we use a mix of commercial algae that you all are all probably very familiar with. The Nano, the Phyto Feast, Live, and the Shellfish Diet, all from Reed, Mariculture, Reef Nutrition. We have started supplementing in some of the TP with some of our animals, and we use a pretty shellfish diet heavy mixture here. Probably 60% shellfish diet, and so we're just using some very common commercially available foods. We have tried producing some algae here in the past, uh, and we purchased a small bioreactor a few years ago from a company out in California. It's not nearly as complex or interesting as the bioreactor that Monte has. But we tried that. We were able to grow Nanochloropsis, and we were actually able to grow one of the diatoms, and the name slips me now. But we were able to produce about 5 gallons of green water a day. But we did the numbers on that, and really the juice was not worth the squeeze on the upkeep and the trying to keep it together. We were really just better off purchasing the commercial foods. Our buildings, we are unique in some ways in that we have a lot of space. We have a mussel building that if you've been to Virginia Tech, if you were to walk in here, you would have some serious deja vu. We also have a clean lab, which is really nice. We have a greenhouse that is no longer a greenhouse. It's been converted to a enclosed building, and it is being used now as our grow out building, and we're working on building that out as we speak. John has been working on that for the last 3 or 4 weeks, trying to increase our capacity there. And then we have our wild fish building, which is majoritively used for some of our crayfish culture, but it's also used for our host fish and some other things. Speaking of host fish systems, we use a pretty straightforward variety of host fish systems. we use the Ahab racks, of course, and then we've used these big round pan systems or tub systems for our largemouth the last couple of years. Um, that pink table system that you can see there, also known as the pink table system. It works really well for holding largemouth and some shiners and things like that. It's nice and round. We've been using that to collect a lot of our Lampsillus the last couple of years. The little trough system that you see on blocks there, we've been using it for sculpting and some shiner production here as of late. It seems to be working really well. Once again, very simple systems that I think most of you have seen or used in the past. We have some collection bag dropouts on there just to put those in, so pretty straightforward. Broodstock systems, even more straightforward. Pretty much these little pan systems are all we're using for broodstock. I think they hold about 5.5 gallons of water or something. We're able to hook those up and run pond water into them or run them recirc. I see that it's cut off the bottom of them here in my presentation, but they have about 100-gallon sump on them, so we can run them recirc if we need to. So pretty straightforward system there, vertically integrated to allow us to hold, multiple animals. Split those out a little bit. I think we have 4 of those right now. So pretty straightforward system there as well. Juvenile culture systems. I'm sure you're going to see a bunch of these today. Very, very simple, straightforward system. The system that Anna and John, I think, put together at Virginia Tech has morphed and spread everywhere across the country. And this is John's latest version of that pulse flow system. Over on the right, I think it holds 21 boxes in that blue tub, if I'm not mistaken. And in total, we have a capacity to hold, I think, 325,000 drop-off juveniles in our juvenile culture room. And of course, that capacity goes down as they get larger. But in general, we've been moving them into these tub dwellers. Once again, John designed these, I think, while he was at Harrison Lake and brought them over here. Very, very effective. We had to manage those a little bit. Those recirc, I think they're about 150 gallons a day. So feeding those and keeping those clean is fun, but other than that, they work really well. And then our final stage of grow out is, of course, the baskets. I think, we really have invested heavily in the baskets and the rings. They're expensive. I think Anna just got some over at Harrison Lake, so that's really cool. Hopefully those will work out great for you guys. Though they really are an investment, but the lifespan on the rings is pretty good, and the baskets are pretty good as well. And so it's definitely worth the investment not to be hunting down pool noodles and that kind of thing. The baskets are just kind of tethered on a line out in our ponds. So pretty straightforward. Once again, we really operated on the do 1 or 2 things and do it really well principle here. We're not trying to be, we're certainly trying to be innovative and creative, but also if we find something that works, we're not going to reinvent the wheel for no reason. One other thing that we do is we tend to, or we are working toward doing some cage culture with West Virginia. We've been doing that for before I got here, 2019. We've continued to do that work. We use a couple local lakes here in West Virginia. We have done a few things differently the last couple years. I think a lot of folks are putting fish into those cages that are infested. We've went around that, and so what we do is we do all the infestations here. We drop the animals off, grow them to a couple millimeters, and then we transfer them to West Virginia to put in the cages. It's really cut down on the work and it's increased survival from those. And so I think West Virginia is pulling around 3 or 4 thousand animals a year out of those cages for pretty minimal effort. So we're really happy with that. We've been doing a lot of propagation with it for Pennsylvania, and I've a couple species that we've worked on here that have been really difficult to deal with. Have been Trigonia and salamander mussel. I'll hit the kind of highlights with that. Uh, we've had problems with pistol grip, trying to get the host animal and the females under the same roof was a lot of trouble. Flathead catfish at the appropriate size are really hard to get hold of. And obviously nailing down when the pistol grip were gravid was a process that we got a lot of help from PADP and from Pennsylvania Fish and Boat on, but we were able to figure out when to get those animals. But getting the catfish was still a big deal. So we ended up, growing our own catfish. We learned how to grow flathead catfish, which was quite the process, and be happy to talk to anybody about that later on. But we're trying to innovate here as much as we can, and really find ways to get host fish and animals under the same roof. Paid off in huge dividends this year. We stocked 10,200 individual Trigonia, which is really cool. And of course, salamander mussel's been an absolute pain in the butt. And we found out axolotls work for that as a substitute for mud puppies. And so, that's been something that we've really been pushing, is trying to be innovative on host animals. When you can't get the wild host animal, or if it's difficult to work with, trying to find good production host species. And so the axolotls have worked out great for us. They keep in captivity really well, and the public loves them. And so that's been a really cool thing that we've done. You can see some infestation photos here. Just for this Pennsylvania project over the last couple of years, we've really, you can see 2019 when I got here and John showed up. We gradually built our numbers up to this past year with the big Trigonia stocking. I really firmly believe it takes 3 or 4 or 5 years to really get your feet under you at a new facility, and I think we're finally starting to stick with that. So that's great, and we've produced a lot of other animals on the side for West Virginia and other folks too. But those are interesting. One thing, the food color treatment. I don't know if you guys saw John's presentation at FMCS 2 years ago. This thing worked out. It's really cool. You can put food coloring in the water, change the color of the animal shell. It works best on lighter shelled animals. It's kind of effective. It doesn't last as long as we'd like. The FDA really got upset about it, so we've kind of put that to the side. We are allowed to kind of do it, but we haven't done a whole lot of it. Future work wise, we're getting ready to work on Pleuromimus clava this spring, short-term brooder, developing fish hose for it, and really trying to figure out how to do it. We did get to see it spawning last year, and nail down the time to collect that animal a little bit. So that was really cool. Research, we're doing tagging studies, food studies, host fish production, trying to bring in some graduate students. Of course, all of our partners, and questions and comments if you guys have it.
We got plenty of time for questions.
Hey, I have a question real quick. This is Tim.
Yeah, go ahead,
Go
Tim.
for it, Tim.
Drew, uh, what are you dosing your young juveniles? I think we've talked many times about your feed mix, but like, what's a daily dosing?
Um, how much or how often or
Oh,
what's your
yeah,
sorry.
yeah, both.
So we feed the boxes generally once or twice a day. To be honest with you, it's according to whether we're on a weekend or we're here. Obviously, the boxes are kind of fed once a day and to allow that food to settle out. The down wellers we generally are feeding once a day. We may spread that out a little bit, especially if the animals are a little older and they're really getting a lot of food. We try to break that up some.
All right, thanks.
Drew, can you tell me a little bit more about your subwellers, like your water flow, like what water you're using, the water flow through there, what your water, what your cleaning and water changes are like?
Yeah, so those are usually on our 5 micron filtered pond water. And that is exchanged. Uh, we usually do a daily water exchange on those animals, usually in the mornings. And those systems are cycled. Usually, I think it's every 2 weeks. John shuffles them all around just to break the systems down and get them restarted.
And what size are they going? What size are the mussels when they go in there?
It seems to vary, to be honest with you, Chris. It's kind of a species-based thing, and to be honest with you, that's been a little bit of trial and error. But in general, it's when they tend to make the switch to filter feeding. We're generally putting them over there when they're holding on about 1,000 micron screen.
Hey, Michael, you got a question?
Yeah, hey Drew. I was just wondering what you were using for your host fish for your round hickory nuts.
So with the round hickory nut, we found that the little fantail darter that works out here or lives out here in the creek in front of us works really well. It looks like genetically should be broken up into something else, and I'm assuming that complex breakup's coming at some point, but not exactly uh, leading the field on fish taxonomy. But the fantail that's out here works for us, and also the sculpin that's out here, which is modeled sculpin, I think, right now, but it also may be moving around, which is interesting because I think Monte may have seen something different on the sculpin. I think that's right, maybe not. But modeled seemed to work for us. We got animals off of both of those. We only did that 1 year due to some permitting stuff and trying to shuffle space and funding. But we're looking at maybe trying that again in the next couple of years to expand that, to see if we could get up to production numbers on that animal.
Okay, it's really cool. That was a species we've talked about doing from in the future, and I know some of the host work utilizes Amacrypta. That's a species we can't really get a hold of large numbers, and they kind of terrify me to try to keep those. And
Well, yeah, I think that's kind of the message that I've been trying to get across in a lot of our stuff is, and right or wrong, we've been trying to find alternative hosts that are easier to keep. Because Amacrypta or some of these other things, mud puppies, if you bring them in and you infest them and they all die at the end, before anything comes off of them, or they die before you can put anything on them. You know, the oxalotls are about a 30% host, right? They're about 30% as good as a mud puppy, but some of them actually live to the end of the infestation. And so 30% is better than nothing right now. And I'm not saying that's a long-term solution, but yeah, for things like around hickory nut, if we can use sculpin and fantail stuff that we're able to hold pretty easily, then yeah, I think it's a much better option.
Good deal. That's what I wanted to hear. Matt, we do the same thing. We use the hosts that survive in the hatcheries. A dead host
Yeah.
produces no mussels, so
Right.
that works. Thanks.
Okay. I'll give an update on kind of what we've been doing, some of the strategies that we're employing here at the Virginia Tech Lab. Just a couple photos of a release here that we did. One of our projects is in the Shenandoah system, and it's a bunch of Atlantic slope mussels that we released. We had a pretty good year on this particular batch. So these mussels were released in the South Fork of the Shenandoah. Just real quick, the folks working there, Bill Henley, who mainly does histology and was also trying to develop some of the in vitro culture. Don, who heads up the Atlantic slope mussel culture. I'll talk more about the setups here. And then Ethan Anderson is a recent graduate student that's also kind of heading up the Upper Tennessee lab and culture work. And then Parker helps him out, but also kind of generally helps out. Alyssa Ganser is a PhD student finishing up her PhD work. And then a relatively recent hire, Ellie Stouch, is helping Don do the Atlantic slope mussel culture work, and then myself. We've had a slew of recent folks in the last couple of years, graduate, in terms of just folks coming through the program. But Zach and Isabel, Kayla, a couple of folks that are on the call. So Katie just finished up her master's degree. Okay, the thing I want to point out, and I know folks have seen, some folks on the call have kind of seen this a number of times, but there are folks on the call that have not necessarily seen some of this stuff. So I just want to point out that this is the main mussel lab here, okay? And then there's the Aquaculture Center here. And so the mussel lab, right now we're doing Atlantic slope work, and then the Aquaculture Center, we're doing Upper Tennessee work. So we've got both of them separated out. The mussel lab here uses the pond to the left. And so that's pumped into the main mussel lab to grow those mussels. And then the Aquaculture Center has a mesocosm. I want to spend some time talking about that. So that's this outdoor unit here. And again, I've talked about this, but I do want to emphasize the effectiveness of it and just how really little space it kind of occupies relative to the pond. Our pond at the mussel lab, I think it's 1/8 of an acre. It's not even a 1/4 acre pond. It's relatively small, but the mesocosm's even much smaller than that. This is what it looks like. There's a number of tanks and things, but the big thing is it's water. It's got flowing components, got a couple pumps moving water around the system. It's getting sunlight. There's fish in the system. We're feeding the fish, and we have been working on this system. In fact, Drew, I think started helping on this, back in 2011, 2012. But we've been, consistently working on this for about 15 years or so, and making steady improvements on it to kind of get it to work. Just a look, you can see in the summer we get some pretty good, algae blooms. It's just a natural algae. We haven't even characterized the algae species that's doing this, but we usually have about 2 months, and that's about it. It's usually like July and August where we get pretty good algae blooms. The mussels will feed on it, shows up in their guts, and they're definitely growing and culturing well when we get a bloom like that. And so you can see here off to the left, the outdoor mesocosm. It's actually, there's a series of pipes. I think Zach had put this schematic together when he was there, but it's like this, the pipes flow into and are pumped into the main lab. I'll show you some, photographs of that here in a second, but the main idea is that you have something that's akin to the pond. It's got different components to it. It can grow, FPOM, it can process FPOM and algae, and it can be pumped into the lab for growing mussels. This is the indoor mesocosm. This has gone through a number of configurations. In fact, we just recently changed this, and I'll show you that. So again, we've got these, the box systems, the flow system, the flow through system boxes that we developed, a number of years ago here, and we still use these. They work quite well. I will say this, the thing that I think we have learned over the years is just improving the overall management of the number of mussels in those boxes. So we when they first drop off, we don't put any more than about 1,500 in the box, and as they grow, we're slow. Probably at 2-3 weeks, maybe a month, we're starting to reduce and spread out and manage the number of mussels that are in those boxes, and then they get down to about maybe 300 a box, and then we'll grow them to about, I'd say maybe about 2 to 3 millimeters. And once they get to that size, then we move them over to the right to the system. We just recently kind of developed this more flow through system that Athan put together. And so we've got 2 of these systems. It's continuous flow, and it's pulsing algae and water from the mesocosm into the system every 30 minutes. So 30 minutes, there's water from both systems, from algae, and then water, the mesocosm pulsed in there. And then it also is draining some of the water out. And this has been super effective. We've had, excellent results with this this year. Here's a shot of one of the boxes. I think this is Actinonice pectorosa here. It's Upper Tennessee stuff. But holding them at fairly high densities, most of these mussels in the 8 to 10 millimeter range. Some are even a little bit bigger than that. And then once they get in that, 10, 12 millimeter range, we're going to move them over onto this indoor mesocosm. So we had a bunch of these upwellers, and we get these algae blooms from outside. Would come into the center. We'd have them, let's say upwellers, and they fed off of it. They did pretty good, but there was some continual management with this. But the issue that we've been trying to resolve and fix, from the get-go with the mesocosm work, is ultimately being able to have productivity through the cold kind of winter months, and so we can grow mussels continuously at the lab. And so just piecing that together, kind of understanding the engineering, all those tanks in the back of the wall there are holding tanks that are communicating with the outdoor mesocosm. So water's coming into those tanks, it's been warmed up, then it's being pushed either to the boxes or the flow through system or this. But anyhow, we ripped out these upweller buckets. We spent a lot of time on those, but tore those out and then put this indoor mesocosm in. That's Parker and Athan there in the middle, so you kind of get a sense of how big it is. But and the tanks against the wall are basically the same, communicating with the outdoor mesocosm, bringing water in, and then being delivered to the different tanks. But the 3 big tanks off to the left, we're really close. In fact, we'll probably start this week. We will bring in cultured algae. I'll show you that in a sec. And we'll put them in these 3 tanks, and then we'll push that over into the system off to the right, which is the tanks that will be holding the mussels. So you can see the upright system, the flow-through system that I just showed you. Juveniles will come off of that and go into the tanks where Athan is standing there on the right. And so there's upwellers in there, so there's gravel bottom in there. In fact, the tanks here, the clear tanks, the long tanks there, we've got gravel bottom, sand gravel bottom, and we have mussels in there even now, and they're starting to do pretty good, starting to grow. The nice thing about the mesocosm is 2 things. One, allows for growth during the wintertime, which is a big thing. But we really have 2 sources of food. We've got the algae that we can produce. Let me show you this here. So this is some of the algae that Parker was an oyster culture specialist in a previous position. So he had learned a lot about how to grow algae. So we're doing 3 species of algae: chlorella, Nanochloropsis, and a diatom phaeodactylium. So those are being grown there. And then we put them into vats, and we're going to be moving them into those, those 3 big tanks so we can get a bloom, during the wintertime, keep that bloom up, and then feed it to the juveniles off to the right. And so we're in the process of doing that now. And I feel like probably in the next month or two, we'll have some kind of initial results in terms of how are things going. But generally speaking, we've been able to have pretty good results with the mesocosm setup, continue to try different things. And we're really trying to solve being able to kind of like grow them kind of year round and just, cut down the amount of time that they're in the lab. And then this is the main mussel lab where we're doing the Atlantic slope work. We've made a bunch of upgrades over here. In fact, over the last 2 to 3 years, I mean, every system that we've had in here has basically kind of been torn out. It's been reorganized. We put, systems, so you can see everything off to the left is for box systems for holding, drop-offs. And then a little bit further down the lab, you can see these larger tank systems that are connected directly to the pond. It's akin to a flow-through system. It's something a little bit different than we're doing over in the Aquaculture Center, but having excellent results with that. We take the mussels, they get about maybe 2, 3, 4 millimeters. We drop them in those big tanks. It's just continuous flow of water from the pond, and we don't mess with them. We don't touch them, basically till like October when we pull them out. So they're in there for about 4 or 5 months. Basically the summer, and we're just, cleaning the tops of those tanks, and that's about it. So a lot of kind of system revisions here. And then just, kind of what we've done, this is for our Upper Tennessee work. These last 5 years of production, we basically work about, 8 to 10 species there. You can kind of see, but we've produced over 658,000 mussels, about 10 species, been pretty productive each year. But I would say that the good thing, especially starting about 2022, maybe more like 2023, the management of the mussels in the boxes, just kind of pushing them, managing through the systems, keeping the numbers low. As long as we do that, we've had really, really nice results. In fact, even though we didn't produce a big number, 104,000 in 2024, we've had excellent overall survival. I don't know what it is, but it's pretty high. We probably have at least pushing about 25,000 mussels from that particular cohort at the lab right now. And then we've got, of course, one of our key collaborators has been Virginia Department of Wildlife Resources on this Upper Tennessee project. And what we have been doing is mainly, propagating the mussels, getting them to a certain size, and then moving them down there. They have, much better kind of more long-term, grow out capacity down there. So all the, Pectorosa, Ligamentina, a few other species that we're working with, we'll transfer them down there probably, maybe late May, early June, and then they'll top them off for like, 3 to 4 months down there, and then, then they'll be released. They'll take care of the release. And this has been kind of a nice, working relationship, just kind of taking advantage of, the things that are good at each facility. So it's been nice to collaborate in that way. Okay, so I think this is more of just mussels that have been transferred, but you can see off to the right-hand corner, as of say late last year, we've transferred over 64,000 mussels, and these are a little bit larger juveniles at 2 to 8 to 10 millimeter range of about, I think, 10 to 12 species of different things over the years we've been transferred down there. And from that, they have grown those guys out and got them released. But working with them, we released almost 20,000 mussels. I think of like about 10 species there. So each year has been pretty productive. We'll continue to do that for the next at least few years. Okay, so switching gears a little bit, this is highlighting some of the Atlantic slope work. This is our production work over the last, say, 6, production seasons. We're pushing almost or over 900,000 mussels that we've produced. And I will just say, for the suite of species that you see off to the left, many of these species we had not worked with, maybe a little bit here and there. And so, there was things about figuring out the fish host and, where to collect them, where to get them, how to hold them in captivity. all those kinds of things. So there's a lot of tweaking, with this project. And this has been a really nice project, to work on these different things that or different species that we've never worked on. We've had some pretty good results. 2024 was a pretty good year for us. We had over 200,000 mussels produced, but of a number of species. I think it's about 10 species or so. And again, through the management of the mussels and getting them into the different systems, I don't know if I've got a slide on this, but I think we're holding over 50,000 mussels right now, between say last year and then mostly from this year, and they're doing pretty well. And then this is what we've released in total for this particular project. We started releasing in 2020, so we've had 5 years of release. Released over 17,000 mussels. We found a really nice spot at one of the state parks, Virginia State Parks, on the South Fork of the Shenandoah, with really nice kind of long gravel bed along the riverbank there, and got a lot of mussels released in there. So hopefully, our early indications from some of our monitoring sites is that the mussels seem to be doing pretty well. And then let's see, this is a total produced for 2024. So between the Upper Tennessee work, the Atlantic Slope work, we worked with 15 species. We produced 14 species this year, over 300,000 mussels. We released 11 species this year, a little over 6,000 for 2024. And then in total, I've been kind of compiling this over the years to kind of keep track of what we've been doing. But we've been producing mussels for 29 years. We've worked with 42 species, produced 4.6 million mussels. We've released 26 species, over 116,000 mussels, been released in that 1 to 3, that 20 to 40 millimeter, size range, 16 endangered species. So it's been a lot of work, over the years, and then just, the steady improvements, just the key steady improvements, and not being afraid to just, tear things out and start over and, really kind of change things as necessary as we see different things that can be done. And Chris, I think that's my last slide. So if anyone has any questions.
Thanks, Jess. That was great. Yeah, I'll open it up to questions. Yeah, Loretta.
Well, hi. I just had a question. I was wondering if you could explain your mesocosm and how your water exchange is going back and forth a little bit more. And if you have bugs coming in and out, harassing juvenile mussels or anything like that.
Okay. I think what I'll do is let's see here. Trying to get back to. Just give me a second here. Sorry.
No, it's all right. Thank you very much.
Yeah. Okay. So I'm going back. So okay. Well, let me just start. I'll kind of start here. Okay. And okay. So this thing that says mesocosm right here. Okay. This is the outdoor mesocosm that I showed you. Okay. So it's got these 3 big tanks. These are 6,000 gallon tanks, something like that. Or maybe there are a few, 3,000, but these are a number of tanks, and the water is basically just recirculating around and around. Okay. So there's, kind of pond-like, areas, and then there's kind of riffle kind of areas, and it all collects in this sump, and then it moves on through. Okay. So, but the key thing is that, it's being exposed to sunlight. So, phytoplankton and macrophytes will bloom in here. Okay. It's also getting, airborne detritus, is landing in here, pollen, everything like that. And it's also getting, leaves and things like that. So there's a lot of stuff that's continually being inputted in there that breaks down. Okay. And then there's fish in the 3 big tanks. And so we feed the fish, and they're excreting ammonia. Okay. So they're pumping nitrogen into the system. And that nitrogen is, of course, fueling the phytoplankton blooms and the macrophyte kind of blooms. And of course, then the breakdown of other stuff. And then that water is being pumped inside. Okay. And so let me go one slide more here, but you see these big tanks on the back wall here. All these tanks are connected to that outdoor mesocosm. So they're bringing water in, but they're also taking water from the tanks that are inside, collecting that water and pushing it back out to the mesocosm. So in essence, it's one big, kind of recirculating system. Now in the wintertime, we have the capacity to shut off the outdoor mesocosm to this system, occasionally bring water in, warm it up, and then, use that water. And then we're now have the capacity now to be able to do productivity, in the indoor mesocosm, during the wintertime. So these tanks off to the left, those are where we'll grow algae. Okay, so we've got grow lights on those systems now, those tanks now. So we'll dump in like a carboy of algae in those systems. They'll be fueled by, some nitrogen sources, and then, the light, and then that will be, grow the algae. And we've already done this. We already know that it works, and then that'll be pushed over to the systems in the right that kind of hold the mussels. And so, again, just emphasizing that they're getting 2 sources of food, the algae plus the FPOM. And Ethan, they're off to the right, he's a master's student with me, but he's going to be, characterizing just the water quality, how the mussels are doing, just the overall, kind of biological performance of the mesocosm, how much FPOM is it producing, what is it composed of, what are, how much algae, the cell density moving through the system, how are the mussels doing, comparing that to winter growth versus, say, summer growth, how the outdoor mesocosm compares to the indoor mesocosm. So, but again, just emphasizing that the biggest obstacle, that we kind of seen over the years is that at a temperate latitude, especially here in Virginia or even further north, is that, you basically have 4 months of good growing conditions. And unless you have some kind of capacity in the wintertime to grow mussels, that can really limit your growth. And so that's the main kind of obstacle we're trying to achieve, getting over that is being able to have a system that's productive, during the wintertime. So does that kind of answer your question in terms of how it's operating?
I think that helps a lot. So then you're not filtering this, you're just taking everything as it comes, right?
So there are filters, so the water that comes from the mesocosm into the lab, just like we would normally have a set of filters filtering water down to 5 microns. We do have filters on that. Okay, we have the standard kind of filters to filter the water, especially for the young juveniles when they're first dropping off. Okay, once they get in that 2-4 millimeter range, a lot of those predators are kind of a non-issue, but we do filter that water just like we would normally do for culturing the young drop-offs. But once they get in that 10 millimeter range or so, then we don't worry about that. The mesocosm system off to the left, where we're holding the larger juveniles, we're not, that water is directly coming from the mesocosm.
Gotcha. Thank you. And then are you supplementing food too, or is it all coming from the algae that y'all are culturing?
So for the young juveniles, when they first drop off, we also use, the suite of, say, reed mariculture out, the shellfish diet, the nano, all that stuff. So we do that. We feed them once in the morning, once in the afternoon, and then once before we go. So they get fed 3 times a day. And then the water in the boxes is exchanged once a day. That works well. It's fine till they get to about 1.5, 2 millimeters, and then depending on how many are in a box. But once they start getting bigger, then they really need that continuous flow. And so that's where we've kind of got that middle step, with the flow through system. And so it's like, then once they get in that range, we transfer them over there, and they're just continuously being fed at that point. And then they really take off, actually. They'll go from 2, 3, 4 millimeters up to like 8 to 10 millimeters pretty quickly if they're being fed. And we're also using, of course, the mesocosm water, but we're also using, they're being fed the reed mariculture standard kind of mix that folks are using. So that's how we're feeding them, but we're going through more algae, because of that, because they need more food. Those mussels are larger. So we're going through, a lot more food. I'll say that. So
when you're growing them in the winter, are you, as you're still pumping all this algae from the inside too, are you warming them, or are they just kind of at ambient temperature? Are they still growing well?
So during the cold months, all the water is warmed up. So we're culturing all the, like everything that you see, that the indoor mesocosm, the flow through system, the box system, all that is about 24 degrees. So everything's heated up. So we try to keep it at a pretty decent growing temperature.
Yeah, well, thank you for my million questions.
Yep.
Any other questions? I know I've got a bunch. We got time for a couple more.
All right, I have just a quick question, Jess. First of all, great job. It's always good
all right,
to
Jess,
see.
I got a couple of questions for you. It sounded like Ethan was going to be doing some comparisons. I guess that's upcoming. I was wondering if you have any kind of anecdotal or any kind of data comparing how you think things are going between your pond and your mesocosms. How do they compare in terms of growth and things like that so far? Do you have a sense of that?
So we've made a lot of improvements over there at the main mussel lab where we're doing the Atlantic slope work. But I would say they're comparable, right? But we don't have the capacity we can grow the young juveniles, that 1, 2, 3 millimeter range in the boxes year round. Okay, but getting to them to that next stage, we don't have the capacity that we do, over there in the main aquaculture center where we're doing the Upper Tennessee work. So my gut feeling, though, Chris, is that seeing what we're seeing now and assuming things work, I anticipate that. I mean, we already have just in those boxes, they're probably averaging 10 millimeters in that box. There's animals in there that are 12 or more millimeters or so. If we get them on the mesocosm with food, I think we can get them to 20 millimeters plus, in less than a year, something like that, or close to a year, and that would be, that would exceed what we're doing, generally speaking. Now, over at the main mussel lab. Now, I will say that there are some species like, Legumia nasuta, that do really well, over at the main mussel lab. And once we get them to a certain size and we put them outside, like Matt was showing, those things just take off. And so, but not everything does that over there. I mean, Lampsilus radiata has done pretty good, but so the species responses kind of vary. But overall, the goal, I'm hopeful that what we're doing at the Aquaculture Center will ultimately exceed, what we're, doing with the standard pond culture. And I know, there's different, system like Matt's system, and then of course White Sulphur Springs has some capacity to do these things, but there's other facilities around the country. But I really think the other thing about the mesocosm is that it just doesn't take up that much space, right? So like if I was designing a place today, it would have, a number of like smallish satellite ponds, but then it would have like a half a dozen or so mesocosms that connected to a main lab. That way I could take one system completely offline, break it down. Then I could have one, system dedicated, for this suite of species or this project. it gives you so much more flexibility in terms of like managing for diseases, managing for different suites of species. If something happens, I mean, there's disadvantages, but I think that the advantages, exceed the overall, being constrained by, say, 1 or 2 or 3 ponds. It's like everyone in this business knows that there's issues with the ponds. It's like we all see them. We can manage for some of the issues, but they also are, they can also be unpredictable in what those things can happen, with the pond in terms of food supply, invasive species, all the stuff that we're kind of familiar with. And so, the mesocosm, while technically more challenging, it gives you, I think, it can give you a lot more flexibility. So.
All right, we got Joe. If you got a question, go ahead. And if I miss anyone's hands raised, just go ahead and speak out. We're all friends here, so don't be afraid to speak up.
Hey, Jess, can you hear me?
Yeah. Hey, Joe. Yep.
All right. How you doing?
Good. How are you?
Doing all right. Doing all right. I was just wondering how many pulses you're doing per day. Are you still doing 3, or are you doing 1?
So for the boxes, when they're young, we do 1 pulse a day, exchange all the water. We do a 100% water exchange once a day. Okay. Now for the flow through system, that water is continually flowing through the boxes at a pretty high rate. We have not quantified the rate, but it's a decent amount of water. I mean, if I had to guess, it probably would fill the box up, in less than in 2 minutes, something like that. So the water's going through those boxes pretty quickly. And what we do is every 30 minutes, there's a dose of algae going into the sumps of each of those systems. So we set it up with the solenoid. There's a refrigerator there that's got a cone in it, that's got the algae in it. We're basically mixing up algae about every couple of days, and then it's just pulsing in every 30 minutes. But we're also getting a pulse of water, coming in from, mesocosm water that's been brought in, been warmed up, from outside, and then it's being pulsed in. And we also have the capacity to bring water in from, the other side of the mesocosm. I mean, the indoor mesocosm that I showed you, and pulse that over to the tanks and then bring that in. So we can kind of do both.
Awesome. Thank you.
Well, I'm Monte McGregor, and I'm with the Kentucky Center for Mollusk Conservation. I know most of you guys. There are a few names on there that I haven't met yet. So if I get a chance maybe to meet you at the FMCS meeting, hopefully here coming up fairly soon. We'd like to acknowledge, we can't do any of this stuff without a lot of partners. And so our staff, funding, all that kind of stuff is very important to be able to get all this stuff done, our support from our families. And we work with a lot of different people and organizations to make this stuff work. Most of my staff, some of them are on here today, but just to introduce some of them, I'm not going to mention all their names, but you'll see them throughout the presentation doing different things. But each one of them kind of have different roles as well. We have a lot of water in Kentucky, so we have a lot of mussels, 105 species, and a lot of rare ones. Pretty much every drainage in the state has quite a few species and several rare ones, so we have a lot to pick and choose from. Unfortunately, we have 29 federally listed, threatened, endangered, so we have to pick and choose and how to prioritize which one to work with and where to work. So we have these areas in the state identified here in the yellow, where we spend a lot of our effort, the green, the Licking, Upper Cumberland, the Lower Tennessee, so on, Ohio. And we have monitoring areas in all of these areas to be able to track these animals that we release and stuff like that. So we have some focused areas that we do a lot of work. And I, have some ranking systems that I use to prioritize how I work with species. And it's based on a lot of things like, how easy they are to work with and how many populations are present. Can you find females? How easy is to handle the host, and can you do it in vitro? So there's a lot of things that go into which species that we work with each year. And hopefully you guys are probably facing the same kind of things, like how do you pick which species to work with? And so this is just one of the ways that we kind of do that. So I'll go right into our demonstration of all the different things that we're doing. I usually take about an hour to do this. So I'm cramming in an hour's worth in 20 minutes, 25 minutes. So be bear with me as I speak probably pretty fast. But if you have questions, feel free to make some notes. Well, anyway, our facility is located in Frankfort, Kentucky. That's our main facility, and I'll go over it separately. We have 2 branch facilities at Minor Clark Hatchery and at Lake Carmelin, which I'll talk about those independently as well. So we do all our intense work at our Center for Mussel Catching in Frankfort about 6 months out of the year, and then everything leaves this facility and goes to one of the other 2 hatcheries. We keep all our brood stock and all that at our other hatcheries and all our juveniles for grow out. So we don't have to raise any juveniles at our Frankfort facility in the wintertime. It's all done at the other 2 branch facilities. Our center is located in Frankfort, Kentucky on Elkhorn Creek, which is Kentucky River system. Is located here because we had a hatchery here built in the 1940s, actually obtained early 1900s. So it's been around a long time, but only since 2002 we're doing the mussel work. We have several buildings. I'm not going to go into each one of these, but basically we have buildings for fish, buildings for algae, buildings for grow out mussels. We have storage for our boats and stuff like that as well, and office space and conference rooms, things like that. We do a lot of things here. In addition to propagation, we do survey monitoring, research management, a lot of education. And of course, we're going to talk about propagation today. We also raise hellbenders here. I'm not going to really talk about that too much. If you have any questions, feel free to contact one of my staff, Travis Bailey. He's on the call as well. You can chat with him. He does most of the hellbender stuff. We'll mention a couple things. Julianne and I have been working on a guide for freshwater mussel larvae for the entire North America. So you guys, if you're handling some larvae from anywhere in the country, if you wouldn't care to share some of those with me, I'll include some of those. And I think I have up to 61 genera or 61 genera that I'm using now in this guide. I've covered every genus in North America. And so I'm going to be working on finishing that up this spring. So here's my contact information, which will be at the end of the presentation, but basically send them to me in 7% alcohol, some closed, some open, and then get them to me, and I'll have some, take some digital pictures and can send them back to you when I'm done with all this. Well, let's get into the mussels. Center for Mussel Catching, we have a greenhouse. This is just one of the things, but I'm going to go through kind of the process of how we normally give a tour, like we intended. So one of the things first is our water. So we have 3 or 4 different water sources, our Elkhorn Creek water. We have well on site. We also haul in Licking River water from our hatchery because it's so easy to deal with. We got a 2,000-gallon fish truck. We like that water really well for some rare stuff. We also have our own, what we call our house water, which we make out of RO water and store it in these 1,500-gallon tanks in our back building. And we pump it underground to our buildings. This is some important things that I think you might want to think about. This is ideal water conditions that we have that we like to shoot for each year. And so this is really important for us to keep these parameters within these ranges and keeping it kind of consistent. So if you have questions about that, just let me know. Well, first thing we do, we take our well water and we strip it down with RO and add it back with some minerals, with some well water, and take some activated carbon, UV sterilize it, 1 micron filter it, temperature control it. And we use that for basically cleaning and things like that. Our tray systems, which I'm going to talk about in a minute, we basically pump our water from our back buildings up to these silver colored tanks in the top of this rack on the upper left, and gravity feed to our systems so we can have water source to all our juvenile mussels. All our sinks are equipped with our house water so we can clean all our mussels at any sink that we have. It really helps us with just maintaining and having a water that we don't have to use from the wild or anything, just to clean our mussels. It's important since this is done weekly. Just to mention a couple things, I've heard people mention solenoids and stuff. These little timer controlled motorized ball valves are amazing. You can set them to be on for 5 seconds and off for 2 hours, whatever you want. They work really good. They're about $40. We have them all over our facility to kind of do our water changes, food changes, whatever they need. They're really, really good and real easy to use. We have a couple of features on our mussel trays. We keep these signaling mats underneath our tanks to kind of keep them warm. Not necessarily to control it completely, but we don't want them to get too cold at night. And then we don't want them to maintain a certain temperature like 24 to 25 degrees C. And this is just an easy way that we can do this without having a thermometer in the water, since our tanks are a little bit, or they're not recirculating. We have back buildings is mostly dedicated to our fish tanks, a couple buildings for that. So you can just see a variety of tanks. So a lot of these are very similar to what other people had earlier. Aquariums, small tanks, AHAB systems, so on. One of the things that I think is very important for mussel culture is to basically have ability to hold your brood stock. Mussels brood in the fall, some species, some in the spring, and you need to have access to your brood stock. Sometimes the rivers don't allow you to do that. So holding them in captivity at one of our branch hatcheries, which is all natural conditions, allows us to bring our brood stock in pre-brooding and let them spawn in captivity, and then we have access to them pretty much during their normal brooding time at our hatcheries. We mark them, tag them. Sometimes we even bring them into a little control system on the bottom right there, where we actually control the lighting, control the temperature, and kind of trick the mussels into thinking that they're in March even though it's in May or June. So this gives us an extra chance to work with them sometimes, and especially for a short-term brooder that are real sensitive, we can bring those in. Generally speaking, we like to use 3 to 10 individuals per year for our brood stock of a species. That's just kind of a good number, and that seems to produce some pretty healthy juveniles. Just to mention, we use syringes with sterile water to get our larvae out. We record a lot of information like site, female, tag number, age, all that stuff. I'm sure most of you guys do the same. We also weigh our animals as well, and then just the general condition of the animals. We estimate larvae using our computer system. We can count a dish of 10, 15,000 larvae in about a minute and a half or less. So this is just how we do that. If you have questions about that, let me know. But this is a quick way to estimate your larvae. You can get tighter precision with more samples, and it takes a couple extra minutes. We have fish infesting area. Just a couple things mentioned. I'm sure all of y'all know how to do all this, but I'm trying to give a tour of our facility. So just an area that's good lighting. I have all the microscopes nearby, your trays, your handling, your gloves, all that kind of stuff. We only pipe it when we use our fish on one side, so that way we can make sure that we know we're not over infesting the fish, which can happen. We try to use gloves when we're not handling the fish to help. And then you can see we're just dripping in the larvae on one side of the gills after they've been anesthetized. And again, this is using very clean larvae that we kind of estimate how many are going on these fish. This is another example of that. One thing we do quite a bit here, I've been doing in vitro culture here for over 20 years now, basically skipping the fish host. We have a lab that we do this in, and it really has a lot of stuff. It's gone through a lot of transitions. I've had some PhD students to help me with this stuff, Christopher Owen. We've made a lot of changes in it, but we haven't really changed a lot in the last 5 or 6 years, other than some maybe new formulas for a few species that didn't work with our regular formula. But we've tested about 74 species, 62 of them have been successfully transformed in the media, 42 different genera. It does take specialized equipment, incubators, CO2 incubators, temperature-controlled incubators, pipettes, hoods, sterilized water, all that kind of stuff. Here's just an example of what that looks like. There's 100,000 larvae in petri dishes ready to go in the incubator. They've been counted and sorted out by species and stuff like that. But just an example of in vitro culture with Cumberland bean. You can see it sometimes transformation varies a little bit when you're testing things and the serum's not right. But once you get the serum right, the transformation can produce you a lot of juveniles, and you can see the numbers down at the bottom. Even from 12 to 30,000 juveniles just in 4, 5, 6 dishes. We've cultured several rare species. Surprisingly, for some reason, I'm not sure why, but a lot of the Epioblasma work really well in vitro culture, which is surprising to me. I thought they'd be the toughest. Some of the more common ones are hard to do. But we've raised a lot of species in vitro culture, a lot of rare and endangered ones, and we're going to continue to kind of do that. Just another example of how we record some of our information. You can see that the serum types change a little bit, and those are just our abbreviations for what kind of serum. We generally use rabbit serum for most of our stuff, but some species won't work in rabbit serum, so we try different serums. Paddlefish work for some species, carp serum, buffalo serum, horse serum. So there's a lot of different kind of serums you can use, and not all of them are equal. One thing I will mention about our algae before we feed our mussels, we have to raise our algae, and then we have to process it, and this is what it looks like in the refrigerator. I've heard y'all mention other species of marine algae everyone uses, TP, shellfish diet, nano. We feed a little over half of our diet is freshwater diet, so 3 species. I'm going to go over that. And we also add this stuff called worm castings, which is basically a source of organic debris and detritus that we found to be fairly important to the food source. And I'll talk about that maybe a little bit later if you have any questions about it. Algae provides the majority of the nutrients needed for mussels. You can see that they provide all the things they need, and they need protein, lipids, carbs, vitamins, and minerals. And here's a chart that I made that shows all the different things that each one of these have. I think a lot of the freshwater mussels need omega-6s, but that's just my opinion on that. But I think they can use the omega-3s that you get from the marine stuff as well. But it provides the right mixture of food. We raise 3 species of algae here, freshwater algae, 2 greens, Neochloris and chlorella, as well as the diatom, phaeodactylum. This is one that we got from Catherine Gattenby years ago, phaeodactylum from White Sulphur Springs. And we added chlorella, which was one that has all the 20 essential amino acids, very nutritious. You can actually take it as a food supplement for people. So it's very, very good food for the freshwater mussels. Again, you got to use a lot of specialized equipment, sterile equipment, and all this stuff to do your algae. We buy our slants from University of Texas. We make our own slants. You can see on the top, these are our own culture slants. And then we put them in these series of incubators from the slants to the small flasks, to the medium flasks, to the carboys. And then we can expand this to larger tank systems, such as 100, 200 gallons, or even bioreactor. The diatoms tend to be cultured in the same kind of way, and carboys, they do a lot better. We have to control the temperature on the diatoms. They can only culture about 18 degrees C. They won't do well at higher than that. One thing with the greens, we have to control with use carbon dioxide, but with the diatoms, we just have to hit the pH really high. So they take quite a bit different conditions to be able to raise them. Once we do have our algae, we don't feed it right out of the carboys. We have to wash it and to basically centrifuge it, reconstitute it with water, and try to get it at a density equivalent to what the nano and the reed mariculture diets are. And those are generally 0.4 to 0.5 grams per mil. This just lets us have a standardized food that we can know we're feeding a certain amount of food, volume, and also cells, a weight of algae per weight of mussels, basically. Here's our diet we typically use. I know someone was saying they use a lot of shellfish diet. That's actually probably the least amount of our food, although TP is a really large component in that diet. So you can see our greens are 6 parts to 1 to 4 parts diatom, 2 parts nano, 1 part shellfish diet to 6 parts the TP, 1800. So again, we centrifuge daily to remove those salts, and then we add a probiotic. I'll talk about that just in a second to give it some consistent bacteria. And then we have other components that I'm going to show you in a second to help with some ammonia reduction. This probiotic we use is called the Clearfo 1002. I've tested many of these. It's got 11 species of common bacillus type bacteria, which is common at all our mussel sites in Kentucky. So that's the reason I use it. It just gives us a consistent source of bacteria to feed these animals. So our culture systems, you can see we have just simple trays. These look simple, but they're pretty complicated as far as the design behind them. But they're static trays. We just use aeration that has oxygen on them. We have the heating mats under them. These drains are just allow us to clean them, and I'll show you that in a second. Here's an example of how we clean. You can see the bioballs that are in the trays. We have a very small sand in there, less than 100 micron sand that's been sterilized. Here's what we generally do. Once a week, they get 3 times the water change a day. They're not flow through. They're just static systems. They get natural light in the greenhouse, weekly cleaning, 10-day counts and measurements, and each tray is independent of the other one. There are about 6 laters in the water. We have bioballs for ammonia control. We have sterilized sand that's cooked in an oven. We add pure oxygen from an oxygen generator. We have heating mats for temperature control, drains for easy cleaning, and then a mobile cart to help us maintain some of our sand so we don't lose it. We also add the food plus the probiotic together, and then we spread that out among all the trays. We feed 1 to 2 meals a day is all we feed per tray, and those are density based. So if you have less than 1 gram of mussel, they're going to get only 1 meal, and if you have up to 10 grams, they'll get 2 meals. We also add the worm castings Monday, Wednesday, Fridays, and that's only a milliliter of that concentrate. Anyway, just to kind of, we prep the worm castings by running it through a sieve, microwaving it, and that's what it looks like in that little jar at the bottom. We pre-prep our bio balls so we can always maintain them and change them out every 2 or 3 weeks. And we sift our sand, so we always have really small sand. We track our juveniles with the batch number. If anybody has a question about this, basically the first initial of the species of the genus, the 3 initials of the species, followed by the year of the culture, the number of cultures. So if we had a 24.5, that means we've done 5 cultures of that species. And M just stands for the media or the fish culture. And we also can identify with the tags. Sand size is important. You can see the difference here from small sand, less than 100 microns on the left, all the way to 200 to 450 microns on the right, and the relative size of the juvenile mussels. So it's very important to characterize your sand. Not all sand is sand goes from 64 micron to 2 millimeters. So it's really important that that you pay attention to what size sand you're using. I think it's very important for some of the mussel species. Also using the proper use of screens. Some mussels will fit through a screen sideways, so just be careful of that. We have all kinds of screens available all the time for different species that we work with. And I will generally use the height to kind of characterize. Like a 400 micron height, we'll need about a 300 micron screen. And just always watch your animals, if you have some questions about this is kind of stuff that Michelle, who takes care of all our juveniles, just looks for things daily. Just, how much food are they eating? Their guts look good. Are they siphoning? Are they crawling around? Just little things, checking the smell of the food, checking the water chemistry. All these are very important things to make sure your mussels are healthy. If you have questions about why we're looking at certain things, I have some literature that I've gotten from the early 1900s. I'll be glad to send you guys some copies of what the people look like 100 years and what they looked for 100 years ago. And it's the same thing. So why reinvent this stuff when they were looking at them 100 years ago? Again, just more things as the mussels grow. You're looking for siphon anatomy, looking behavior on the foot, looking for odd things like stuff growing on the shells, predators, competitors in the tanks, insects, midges, and so on. But here's what they look like in these little trays. There's hundreds of mussels in these trays, and just little piles of pseudofeces next to them. But generally speaking, we get fairly good growth in about 3 or 4 months. We get 4 to 5, 6 millimeters up to 10. And it is important to kind of the density that you use. Here's some that are just a little bit older, 20 weeks old, but yet they're a lot bigger, and it's because we kept the density down. So in general, what kind of growth we normally get, the first 3 months, 12 weeks, we're getting to 3 millimeters. And generally speaking, for most all our mussels, if they're growing about 1 millimeter a month, then we're happy with that at 24 degrees. If they're not growing that, then something's not right with their food and something's not going on. Survival for 4 to 6 months is pretty good. But I think anywhere from 3 to 25 to 30% is normal. Sometimes you'll get lucky and get 50% survival of juveniles. These are transformed juveniles. But generally speaking, if just kind of, we always plan on 10%. Just plan on 10%. If you get more, that's good. If you don't get 10%, then you need to have a couple batches to get your 5% levels. Just another species of Aquata. Again, variation in survival a little bit, but still you're getting quite a bit of mussels if you plan on your 10% amount that comes off. The mussels, once they are 3 to 5 millimeters, they leave our facility in Frankfurt, and we transport them in these little silo cups. It's really easy. We can put 500 in these little cups, put them in a big cooler. We might have 100 of these things in a cooler. Or whatever will fit. We aerate that cooler, we transport them, and then they're ready to go straight into the next step, which is the basket. We also weigh some of the animals so we can see how much they're weighing. How many do 500 of them weigh? How much do they weigh? So that way we can track their growth not only in length, but also in weight. Just to give you an example, the mussels of Trustensis on the very top there, leaving the greenhouse about 4 to 6 months. We had about anywhere from 62 to 500 per tray, 4 to 7 millimeters. About 3 to 7 grams of mussels per tray, or about a little bit less than a gram per individual. And that was very similar among a couple different species. So we're raising these things at densities of between 900 to 7,500 per square meter, or 3 to 9 grams of mussels per about 6,000 grams of water, or even more fine, about a half a gram to 1.4 grams of mussel per liter. So if you have questions about that ratio, let me know. But here's the kind of growth we have. This is the same species over the last 4 years. We see the very similar pattern. We see a similar growth. We're trying to get them to 3 millimeters minimum at 3 to 4 months, and that way they can be transferred out of our facility into our other. Once they grow another year, they're going to get anywhere from 20 to 25 millimeters generally for this species. Well, they get moved to, how am I doing on time, Chris? I'm not even paying attention to the time.
Let's see. I think you're doing all right. You've got a timer up in the top that you should be able to see.
Yeah, I don't see a timer, so
You've been going about 25 minutes so far.
Okay. All right, well, I'll hurry then.
You may have some questions, but yeah, we're also a little ahead of schedule, so there's a lot here. But yeah, go for it.
Okay, I'll go through these last pretty quick. So here's our other branch hatcheries. Minor Clark is basically a big raceway that we have water from Cave Run Lake, and we put mussels in these baskets. We also hold a brood stock in these, so it's pretty simple. And we'll skip that. Wolf Creek National Fish Hatchery is our other facility. We have a floating boathouse up in Lake Cumberland. Here's what it looks like. We've added this rack underwater. So instead of having cages on these floats, we just still have some of those. We just basically lift all the cages up out of the water and able to get to them without having to get in the water. So we really like this. We also use a lot of these floating baskets. Any kind of floating basket works. And this is just the ones that we have. We also do the cage culture. We have a few of these as well. We started doing it with these, but then we switched over back to the other one because it's easier for us. That's not something new. This was developed in the early 1900s. These cage cultures are something that we just patterned off somebody did 100 years ago. We started in 2016, and then we've got about 39 cages now and 100 baskets that we have currently. We have a little more capacity than that. But basically, after about 15 months, so basically we raise them at our facility in Frankfurt for 6 months or less, take them to there, let them go another year, and then we pull them out of the cages or out of the baskets, run them through these screens, and they're ready to go out in the field. We sort them and tag them. And then here's the kind of growth we get for the 17 months, anywhere from 15 to 30 millimeters, depending on the species. And sometimes we might keep them another year if we feel like they're not big enough, and then they'll get over 30 millimeters, at the third growing season. But again, this is no cost to us. There's no electricity and all that stuff, so it's pretty easy to maintain them for another. And the survival once we leave our facility has been over 95% almost every species. So once they get to 5 millimeters or so, we take them to our hatchery. We put 500 in a basket. We'll get 495 or so out of that basket in a year from that. Just to give you some examples, original 100 mussels at 6 months weighed 3 to 4 grams per 100, and then at 18 months they weighed 70 grams. So that's how much growth that they're adding on. And the densities in these baskets are anywhere from 1,600 to 2,000 per square meters, and that's about the same for the cages. A little bit higher for the cages than the baskets. Again, the same kind of thing. If you have questions about some of these, a very similar survival. It just shows you how we weigh them to kind of track their weight. It's what they look like. The growth is pretty good. And then, we're getting stockable. So what we're doing, we're taking the annual ring of the first year in the wild mussels. We're looking at the second ring. There's our stockable size we're shooting for. Sometimes, we can go to the third year. But basically, that stockable size anywhere from 13 to 25 millimeters, and that's kind of the size we're shooting for to be able to tag them and release them. This is just 23. I didn't put 24 stuff on here, but we had 22,000 mussels, several species. We released a very good size mussels. We have volunteers help us tag these, and we maybe tag 6,000, 8,000 mussels a day with about 15, 20 people. And here just shows you how that works. And we've propagated over 76 species, 62 of those been in vitro, some of them fish, 16 TNE species. And I think that's my last slide, and I got 4 minutes for questions. There's my contact.
Thanks, Mike. That was
sorry, I talked awful fast. I'm sorry about that.
You had to. That was so much. That was so much information. Has anybody got any questions?
Monte, where do you get your volunteers from for the tagging?
We have lots of people that want to come help us do mussel conservation. So, we have a TV show, magazine, and sometimes I do, announce it on Facebook. We have a Facebook Kentucky Wild program. I don't have a problem getting people. I have to actually say I can only have 10 people come or I'll have 50 people. So we have a lot of people that just want to come help us for a day. You just send the word out, and then keep up a list of people that that are interested, and we put it out on Facebook sometimes. Sometimes we have a special event where we'll have, okay, come help us tag mussels on this day, and then sign up to do it, and then we'll have people sign up to do it. And I've had people drive 3 hours away just to come help us tag mussels. So
wow, that's really cool. Thanks.
I've got one question, Monty. I was curious, at what sizes are you, like if for mussels you do later in the year, at what sizes are they going to the hatcheries? Are you getting them to 2 millimeters and shipping them off? 5 millimeters? What's kind of the smallest size that you would feel comfortable sending them to the hatchery to spend the winter in those baskets?
that's a great question. And the answer to that is we've tested multiple sizes over the years. And so what we found is our Lake Cumberland hatchery has about 200 days of growing season. Our Minor Clark hatchery has 160 days. So depending on the time of year, let's say I'm doing something in June or early July, some of the short-term brooders, and I still want them to 3 millimeters. That's 3 to 4 millimeter size range seems to work good as long as they don't go into the hatchery past, let's say, October 15th. So what I want them to have at least 1 month of growing season. What I mean by that is above 18 degrees C before they hit that cold water. So if it's mid-October and it's 22 degrees or 21 degrees, it's usually a lot warmer than that in Lake Cumberland, then I'll know I have about a month and a half left before the early November that they'll still grow a little bit more. They'll slow down, of course, as it gets colder. So I really want them to be 5 millimeters before wintertime. And that seems to be a good size that they won't get blown out of any tanks because they're heavy enough, but they're also got enough fat reserves to be able to make it through the winter. Smaller than that, we've had some 1 or 2 millimeters that we put in, and the survival hasn't been there as well. So we want them to 5 millimeters is ideal. 3 would be the minimum. And again, give them a little bit of time to grow. Don't put them in middle of November. That's not going to work very well because there's not much food left in your systems anymore. Does that help? Does that answer that question?
Yeah, that's great. One quick question, Joe.
Hi, it's Rachel. I'm hijacking Joe's computer. Hello, everybody. Here, I'll even show my face.
Hey, Rachel.
Hi. I was wondering, Monty, I enjoyed your presentation, so thank you for that. How are you weighing your mussels? How are you getting rid of the water? Are you trying to get rid of the water, or are you just weighing them, just dumping them into a pan and weighing them? I guess I'm kind of curious about that because that's the direction I think we should all be going in. And I don't know if anybody else is on this call is weighing their mussels, but I'd like to kind of understand that a little better.
Yeah, if they're 1 to 2 millimeter mussels, you're going to basically put them into a petri dish. Pre-weigh the petri dish so you have a 0, and then you pour all the water out. Take a little Chem wipe or something and get as much water off of them as you can to get a fairly accurate weight. Once they get up to, let's say, 8 or 10 millimeters, you just basically 0 your bucket and put them in there and pour off as much water as you can. And the little bit of water that's left is not going to make that much of a difference. When you have 70, 80 grams of mussels, 1 gram of water is not going to interfere with your results too much. You can also, we do some weighing of individual species. So we can actually, we'll weigh one individual, have 15, 20 replicas of that. And then we'll weigh 10, we'll weigh 50, we'll weigh 100. So sometimes we can just weigh the whole thing and we can guess how many are in there and get fairly close. So it's just a correlation relationship that we're trying to establish. So sometimes we'll just take a subsample of 100 and weigh them. Sometimes we'll weigh the whole amount, and we'll see if we can calculate from our subsample if we can predict how many are in there based on the total weight. So it's just something we've been looking at because we'd like to feed about 10% of our body weight, wet weight, with the food. So if we have 100 grams of mussels, we're going to feed about 10 grams, 5 to 10 grams of food. That's how much they need with our systems. Now, if there's flow-through systems are going to be not as efficient, so you might have to feed more, but our systems are really efficient. We don't lose any food in our systems at all. because we feed them and wait 8 hours before they get a water change.
Hey, Monte, is that dry weight when you talk
No,
about
that's wet
food?
weight. Wet weight, but you can, if you want to do dry weight, it's generally about 8% dry weight is what we normally find. We have done some dry weight tissue analysis, and so it's about 8% of your wet weight is dry weight, and then about 10% is kind of what we keep for our food. Our food is wet too, so we don't dry our food out, and we can do the calculations and stuff, and we've done it, but generally speaking, the wet weight of the food and the wet weight of the mussels is kind of what we're looking at.
So is that like paste after you spun it down and you've got a paste, you're using that to weigh?
Well, I mentioned earlier in our, how we process our algae. So we're standardizing all our food. So we know we have 0.4 grams of food in 1 milliliter. So that way, when we're feeding 10 meals, we know how much weight is in that food because we've already pre-weighted. Gotcha. So we have a pellet of food. We mix it with the X amount known of water. So when we're feeding all the volume, but we're also feeding a known amount of weight. Does that make sense?
It does. Thank you.
All right. Thanks, Monte. I guess we will go ahead and move on now to Harrison Lake.
All right. Hey, guys. Welcome to the presentation for Harrison Lake National Fish Hatchery. My name is Anna Dellapenta. Our project leader out here is Rachel Mayer of the U.S. Fish and Wildlife Service. Our current staff is Jacqueline Zelko, Jace Nelson, myself, Joe Nichols, and we just hired Nick Barnes. All right, so our program began in 2007 with Brian Watson and then project leader Michael Odom. We are the Virginia Fisheries and Aquatic Wildlife Center at Harrison Lake National Fish Hatchery. So that's VFAWC at HLNFH. We're a blended family out here of U.S. Fish and Wildlife Service and DWR, and also Conservation Management Institute, which is sort of partnered through DWR to hire the state employees like myself out here. We also coordinate work with local tribes when possible. Our primary role is propagation, but we also participate in surveys, monitoring, and applied research when possible. We're located in the historic Tidewater Plantation area of Virginia, about 22 miles southeast of the heart of Richmond. The hatchery was originally established in the 1930s to raise warm water fish for stocking in lakes and ponds. Our 444-acre property is largely wooded with a 90-acre impoundment called Harrison Lake, positioned in the northernmost section. You can see the lake and the ponds in this shot, which is kind of nice. Our hatchery ponds have a surface area of between 1 and 1.25 acres each, and they range from 4 to 6 feet deep. They're gravity fed by a nearly 3/4 of a mile long canal that flows from the lake. Of the 22 hatchery ponds we have available, we're currently using the 4 in the top right there for the mussel program. We have tentative plans to expand into more this coming season. All right, so our water is naturally pretty soft and tannic. Our total hardness is around 20 parts per million. Last summer, we experimented with adding agricultural lime to one of the ponds. That boosted our total hardness up to about 60 parts per million, and we saw some promising results from this trial run. This season, we're planning to expand our use of pulverized calcitic agricultural lime to more ponds. We're also considering some fertilizing options to increase our productivity. And now we focus on Atlantic slope species here. Since the mussel program began, we've propagated over 14 million mussels and stocked over 300,000 in 18 rivers comprising 6 different watersheds. Additionally, we've provided over 500,000 mussels to 13 different states for research, methods testing, and tox studies. We also partner with VCU and several tribes to raise herring, which we release into a trib to the James and also the upper Mattaponi. And with that, I'm going to hand you over to Joe Nichols.
Hello, everyone. My name is Joe Nichols. I am a biologist with the U.S. Fish and Wildlife Service over here at Harrison Lake. And so I'm going to kind of start us off with how we hold our brood stock. So this is our short-term brooding trough. You can see Rachel over there siphoning out all the fun conglutinates. So it's a recirculating system with filtered pond water, and we use it to hold our brood stock for our short-term brooders like Liptio campanata, Parvospina colina. And so there's a pump that's circulating water from that sump underneath. And then we have all these individual AHAB tanks that we use to kind of keep our brood stock separate so we can keep track of our batches. So it's really useful for, again, siphoning out all those conglutinates kind of individually. And then these are our mussel fridges. So the one on the left is the one that we've been using for quite a while, and we're expanding upon that. And these are to hold our brood stock. And as you can see, there are 3 sumps in the one on the left, and we're going to expand that in the new one. And that's really important for us to keep our brood stock separate. As Anna showed you previously, our map of kind of where we do stuff. We work in the Delaware River all the way down to, say, like the Dan River. So it's really important that we're able to keep our brood stock separate for biosecurity reasons. So this mussel fridge, we can lower the temperature down to 7 degrees, and we do water changes on it twice a week, and it's recirculating. And so it gives us a lot of flexibility in terms of when we do stuff. So it's great. Next, I want to talk about some of our recirculating infestation systems. So this is what we call an RPS, recirculating propagation system, and it's again really great. We have a sand filter, we have a 100 micron cartridge filter, and we have carbon filters, and then there's a lot of water. So we can really maintain good water quality while holding a lot of fish. We can hold around 600 largemouth bass in here, and get up to 90,000 drop-offs. So each of these round tanks, this one that I have on here right now has white shiners in it, but there's a double standpipe, so we can pull that inner standpipe, flush the tank, and flush all the newly dropped off juveniles into these bags that we can spray out and count. So again, really easy to maintain system generally, and it has a pretty small footprint for the amount that we're able to put in there. And here is another one of our recirculating infestation systems. This is, it's a typical AHAB, but we've made a few modifications. So we do have flexibility in how many tanks we can put into one infestation. As you can see, we have all those tubes, and they're behind. We can control which AHAB tanks go behind which tube, and those tubes end up going where you see those bottom holes. We normally put screens, and that's where we'll collect our juvenile drop-offs. So now moving out to our outdoor flow-through systems, we can't really control the temperature. It usually is somewhere around 17 degrees. But we can do a lot with this. And again, it's flow-through, so we don't have to worry about filtration as much. And these are 2 of our bigger tanks. We mainly use them for our LY floater, Eterbacchiana implicata, because our hatchery is located pretty close to a herring run, so we can go collect our blueback herring, and we can fit around 350 in each one of these tanks and collect anywhere from 500,000 to 1 million drop-offs, which is really cool. So, and then you can see on the far left, there's an external standpipe, and that's kind of how we regulate our water level, and that's where we put our bags to catch all those drop-offs. So now, for most mussels that we have, for most species, they'll come inside into one of our indoor culture systems. So this is one of them. This is the pulse system that Jess and Drew both talked about. And it's a similar concept. Everything gets a water change once a day from you know, 5-micron filtered conditioned water that's, you know, set to the right temperature that we need for all these boxes. And each tank gets fed once or twice a day. There's a sediment change once a week. And we can regulate the food amount based on mainly the condition of the mussels and when we do our weekly sediment change. And so each one of these blue sumps can carry, I think around 18 tanks, and we can stock about 1,500 mussels in each one of those tanks. So we can stock around 180,000 mussels in this whole room at one time in these systems, in the vertical and those other systems. So that's really great for us, and we can move mussels out depending on just how we space out our infestations. They can reach a size where we can move them out into another system and, you know, put new boxes in with new mussels, so we can kind of really maximize this space, and it's really great. And then our other system that we have inside is our little sediment buckets. These are fairly low maintenance and not super complex. We have it's just sediment in the bottom of this 2-gallon bucket with an air stone, and we put filtered pond water in here down to 5 microns, and we'll feed once to twice a day. And so we'll do a sediment and water change once a week. And we can this is normally for like our less sensitive species, and we can fit around 42 or more buckets, so that brings us to about 126,000 mussels that we can put in here, which is really good. Especially some of the species have generally pretty high survival, so really happy to have this system. Yep. And then these are our raceways. They are not in the building that they're normally in, because we're actually doing a pretty big drain project. And they are flow through with our pond water. We have pretty productive pond water, so we filter it down to around 300 microns. And we can actually take the juveniles that we collect from the alewife floater infestations. We'll do a volumetric estimate, and those guys can actually go directly from the fish into these raceways. And this is just a cool video I took. We were about to hand these off to our partners, and we took all the sand out that we have that they sit in. And, that just kind of shows you the scale of what we can produce for those alewife floaters. So that's really good. And this is after about 2 months of growth, and they were somewhere between 10 to 15 millimeters. So really great. Next, I want to kind of move back inside, look at some of our intermediate grow out systems. These are for mussels that are just coming out of our pulse flow, and it's essentially a recirculating upweller. And generally, the growth is really good. We can see mussels double in size within around 2 weeks. They are getting kind of constantly fed by our feeder, and I'll get to that in a minute. And we have to do weekly or not biweekly. Have to do water changes and scrubbing everything twice a week, and then we spray our screens just because these cups will end up popping out. So in terms of survival and I think maintenance and how we can feed them, they're great. It's just the footprint for the small amount of mussels we can put in there it's kind of a tough pill to swallow sometimes. So we're working on kind of expanding this kind of space in the future. And then this is a downweller. This is actually really, really similar to what John and Drew had. And it is great. I think the only issue with it that we have is that the mesh size that we are currently using is 1,000 microns. And I think mussels could probably be moving in here a little sooner. But you can fit a lot of mussels in this system, and it's you can again have the feeder kind of constantly feeding it. So this is just kind of a little look at our feeder. We have a timer outlet that these solenoids are plugged into. These solenoids have an Nalgene bottle on top, And our mix that we feed for everything is a mix of shellfish diet, nano, and phytofeast. But for these, we typically have a 3-to-1 ratio of what we put in the feeder compared to what we put just directly into the sump. So we'll feed the sump in the morning, and then we'll put algae in the feeder with some water, and that'll feed the mussels throughout the day. And so having that kind of system where you can constantly feed everything is really important for growing your mussels out. And then I'd just like to talk about our pan system that we have outside. They are, and it's kind of a misnomer on my slide saying it's outdoor, because it's really in our shad house, a brick building that we have. And so we're filtering this pond water. We're pumping it from the pond, having it filtered to 300 microns. It's going, flowing over these mussels in these pans and then being sent straight back to the pond. And these work great because of the high flow that we can get with these. And the mussels can kind of be a little more comfortable in their kind of somewhat more natural kind of environment. This is usually our final place before we release for a lot of our more sensitive species that can't tolerate being on the ponds just due to heat stress. And I think the flowing water really helps with that. On the right is our pan system that's typically more traditional. And then we have our bigger feed tub system, our SFUs, and they serve pretty much the same purpose. So then a lot of those animals that can tolerate being on the ponds after they've been through multiple systems, or sometimes we'll just directly put them out here depending on how big they get in the raceways or in the pulse flow. They'll come out here into baskets. We are trying out those rings that you have at White Sulphur. I think those are going to save us a lot of work because you have to replace those pool noodles. So having something more durable is definitely going to be great. And then just some more construction stuff with our basket. We rivet this black mesh on the bottom, this plastic mesh, and that's just to ensure that mussels can't escape through the bottom. And then we put another mesh layer on top and put sand on top of that. And then we put all our baby mussels in. And then we have multiple lines in all of our ponds and stretch them out. And this is where a vast majority of our mussels go, especially our more common species that can really tolerate, being in this slightly, lower flow, warmer environment. Now I'll give it back to Anna.
All right. So we do a lot of batch tagging here because we do produce so many mussels. By batch, I mean non-individually identifying tags. I know a lot of labs are using black blue dots. We're kind of still evaluating that option. Previously, we've used hole punched hull print material. You can see in that photo. It does not punch clean. It takes a long time to punch them all. And it's just, we needed something better. So a stroll through the craft store, and we found 5mm sequins that have like a slight curve to them and a hole in the center. They adhere very well to the mussels with a very, very small amount of glue. Also, they're available for 99 cents for like over 2,000 of them. So they are, they're cost effective, and you don't have to waste time sitting there hole punching leftover hull print material, which is very nice. We also do something here that maybe not everyone has. We have a laser engraver, which is nice. It kind of blends batch and identifying tagging by allowing us to apply individual markers to many mussels at one time. You can see Jace up there in that photo arranging mussels on a tray to go into the engraver. If you're doing mussels that are all like sort of the same size, you can do up to 2,000 in a day with the laser engraver, which is really nice. After the laser burns off the periostracum layer, we then go over it with a very thin layer of clear nail polish just to add some protection. And also, of course, we do use hull prints. I mean, I think everybody does, so I'm going to spend spend any time beating that dead horse. And also we of course we do pit tags here. This season we incorporated the 12 millimeter plastic pit tags as well as the glass. We're gonna see how they go with our monitoring efforts in the next few seasons. Nice thing about the way we do pit tags here, we use just a tiny little dot of super glue gel and then encase it in Gorilla Glue. We don't use the dental cement. Gorilla Glue seems to be sufficient. Creates a nice smooth layer. You can do it with a small amount of glue. And also, you can buy Gorilla Glue anywhere. Dental cement can be, can be a little tricky to get your hands on. So yeah, that's what we're doing with tagging. We'd like to, acknowledge and thank our partners, and of course, all of our wonderful staff out here. And, thank you. And yeah, questions, please come visit. That's our lake. It's open 6 months out of the year.
I've got one about your laser engraving. I'm curious, and maybe Rachel and even Brian can speak to this. I'm curious as to how long you've had laser engraved animals in the field and how those tags are holding up, over the years, how much erosion you're seeing, and if you're seeing different erosion in different habitats, water hardness, pH, etc. Yeah, if somebody could speak to that.
Brian, I don't know if you want to hop in. Brian, you've done a lot more survey work, but we started using the laser engraver in 2014. There's been a stream or two where we found some erosion on the shell, but we were engraving a little too deep at that point. We're still kind of trying to figure it out. So there is like a sweet spot where you don't want to engrave too deep, even though we do put a clear coat of nail polish on the mussel to try to prevent that. As far as like different sites with different water quality parameters, I couldn't tell you that, Chris.
How long have you been
Chris,
doing the nail polish?
Since we started, Rachel Hoke mentioned that from North Carolina when she was doing that there.
Okay.
Yeah, Chris, some of the laser engraved animals, I mean, we were out in the Nottoway this past year. I want to say we found some from 15 and 16 maybe that we had released out there. So we've seen, they've been out there quite a while. And I think it depends on how well you get that original laser engraved tag on there. Like Rachel said, it took a little bit of some trial and error to figure out, your speed and power that you want to get that set on based on the shell thickness. But some of those look really good, almost like they just came off the laser engraver. Some other ones are a little bit dicier to see. Sometimes you do get some erosion going on in between the numbers, and so it kind of all blends together. But I think that gets at what Rachel was saying about some of those were a little bit too deep, and then the wear started happening, and you could tell they were engraved. You might have been able to see like the letter on the front side or a couple numbers on the back end, but couldn't make it all out. But once you kind of get the idea of what you're doing with each of those different species, I think it definitely lasts quite a while out there in the wild.
Yeah, and I want to point out too that some species are not probably a good fit for the laser engraver, like Glasmeganus beardus. It's your real thin-shelled animals. The laser does get hot. It's burning the shell basically. So if it's a real thin-shelled animal, you probably want to enter with abundance of caution on that. We do engrave implicata, which is pretty thin shell, but not really recommended for thin-shelled animals.
Nice, thank you. Anyone else?
I do want to add. I don't know if I mentioned this, but for our raceways, when we're propagating or culturing our alloy floater, we will also like turn off the raceways for about an hour, and we'll supplement with that commercial algae mix. So that is definitely, somewhat of an important element of that I forgot to mention.
Very good. Well, I am Michael Buntin. If you do not know me, I work at the Alabama Aquatic Biodiversity Center with Dr. Paul Johnson, who I'm sure will chime in at some point. And I'm going to give you sort of what we do for a typical facilities tour, walk through all the steps. Like some other hatcheries have done, here's a list of species we've worked with and propagated over the years. It's about 40 species total, mostly unionids, a couple of margatiferids, and I have to acknowledge our partners too. I mean, we did get a couple of these new transformed juveniles, particularly margatifera like Himbali, Louisiana pearl shell. Those came from Louisiana, and we've been doing a lot of work with Tim Lane in Virginia, and he was kind enough to gift us some lemiox a few years ago. So we've been very appreciative to all our partners. But most of all these other animals were propagated here. And to break down some of the releases we've done over the past few years, this slide does include some snail numbers, and I know this is a mussel talk, but I feel it's important to mention it too because they're important as well. But if you focus down here, we work at a lot of watersheds. I think Paul was talking to the numbers yesterday. We've actually stocked mussels into 20 different water bodies across the Southeast, mussels produced in Marion, Alabama. We focus on a few major ones here. You'll see a lot of stockings in the Locust Fork drainage. That's a watershed that's been severely depleted through years of coal mining and other things. So we're trying to bring some of those mussels back. It's a beautiful river, though. Also, stockings in the Cahaba and the Little Cahaba, spaces we go normally. But in the Mobile Basin, we've stocked just over 75,000 mussels over the years, and over 90,000 snails. We move up to the Tennessee drainage, and you can see the two drains where we primarily work, the Paint Rock River and Bear Creek. And Paint Rock is well known to be a great river, and a lot of it's a source of quite a few of our broodstock, as well as a repository for some of these animals we're trying to reestablish throughout the length of the Paint Rock, which was altered from years of Corps of Engineers activity, straightening channels, removing snags. But some of those habitats are not that bad, and we're trying to reestablish some of those animals throughout the length of the river. And very similar to the Mobile Basin, over 75,000 mussels stocked over the years. That brings our total to around 150,000 mussels stocked. And if you've never been to our facility, we have a massive facility. We're the proud recipient of an old Fish and Wildlife Service hatchery, the Southeastern Fish Cultural Laboratory. It was deeded over to us, the state, in about 1999. We started renovations in 2005, when we had our official grand opening in 2010. You can see some of the buildings, and this is not everything, but this is the primary facilities. The upper right, you can see our wet lab and also the aeration tower. And that's the recipient of two different wells, one of which was drilled last year. It aerates all our well water before it goes into our tanks. You can see the snail pad, open air, good for growing algae and stuff on the bottom left, and our little mussel culture pavilion, which used to be an old pole barn that we walled in and added a whole bunch of drain lines and stuff to. It's where most of the fun stuff happens. And important to know, all the water that comes out of these buildings, the wet lab and the mussel culture pavilion and snails, all goes through our effluent capture system, which is a drum filter system. It catches every particle above 80 microns, and that moves that into our wastewater pond, which is chlorinated with domestic water. And so we're trying to keep the animals on the facility as best we can. Unlike a lot of facilities, we don't reuse our broodstock, hardly ever. We go out, we start in February, and then go all the way to July with our broodstock acquisition. And these species I'm listing here, just the ones we do most years, except for margatifera. We haven't done margatifera in a few years, but it's our earliest brooding species. And we like that approach. It just helps for genetics, as long as we're able to get animals reliably, and that it can be a problem at times. That this is a method we like to use, but we're doing our own field work too, so it's a big task. And very very oddly, apparently, a lot of other facilities we don't hold our brood stock in recirculating systems. All our brood stock is held in incubators. They're held in just simple Tupperware pans we get from Walmart, little 2-liter pans. We like this approach because it allows us to isolate broodstock from each other. If we're bringing in animals from all over the state, all over different river drainages, we don't want those animals contacting mussels from other drainages. Even if we don't know, we don't want any chance of spreading any diseases, whether we know about them or not. And all this water that comes out of these things, we do daily water changes for these things. It goes into our regular sewer, so it doesn't go. It doesn't contact mussels from either the ones we put out in the pond or the juveniles. It's completely separate. And then we're going to return those animals once we've got the Leachidia we need, and we'll take them back to where we captured them with a tag. We hold those animals about 10 degrees, continuous aeration through a little luff pump. We do not feed them. They seem to do just fine, and we've recaptured broodstock year after year with their tags. We've seen them do well. It seems to be a pretty good system for us. We try not to hold them more than 2 months. I mean, in weird scenarios we can, but as soon as we use them, we prefer to get them back out. And that's mostly for our long-term brooders. For our short-term brooders, we changed up a little bit. We still use this same incubators. We don't keep the temperature so low. Extreme conditions when we're getting animals down here in Alabama are probably a bit warmer than everybody else. Usually we're getting these short-term brooders, these pluribemas and fusconias, when the water temps are right around 20. And what we'll do is we'll just simply set the incubator at the stream temperature. And then by the time we get back with the animals, the water's, it's the same temperature. And what we're going to do is bring back stream water. So we found that in order to prevent abortion on some of these animals and prolonged development, we'll bring back water from the stream. 5 or 10 gallons is enough. We'll do our daily water changes with that water, so we don't change water chemistry and we don't shock these animals and cause them to release prematurely. Again, it's the same thing, except we keep one animal per tray this time because we're having loaders. We want to make sure we know who released. We don't want to have to mess with them more than we need to. Provide continuous aeration. Again, changing water daily with that water from the stream. If the animals go longer than that and they haven't released, we can transition them onto our well water, and they seem to do okay as long as it's slow. We don't try to do any rapid changes, and then usually, hopefully, the animals have released at least a concludent or two, so we can see where the kitty are at. And once we can tell they're fertilized, and they're getting ready, we'll start bumping that water temperature up about 0.5 degree a day, every day or two, and just slowly transition them to about 22 degrees, which is kind of our room temperature, and then we can move them out on the lab bench. And usually after a week or so, we'll get a release, and hopefully everybody's good. Hopefully everybody's fertilized and developed, and we can utilize those Leachidia. Okay, and for our host fish, we produce a few of our host fish, most of our bass. We don't use largemouth bass for our host. We used to use our red-eye bass, a couple of species, the Cahaba bass and the Coosa bass. They hold really well in our tanks, and like people have mentioned before, if your fish died, it's a bad deal, your mussels die. So we really like holding those red-eye bass. They do really well. Bluegills, of course, but all of our darters, all of our minnows, those are all wild collected. All of our fundulus, our studfish, those are wild collected, and we're getting those animals from streams with few or no mussels. So we hopefully don't have anybody coming in with them, and we don't have a high rate of natural immunity. We do a little bit of host fish treatment. We don't hold our fish very long. For most fish, we want to infest them within about 24 to 48 hours after we bring them on the facility and get that transformation process started. oftentimes we'll chill them down just a little bit because our temperatures are a little bit higher. We're just in our wet lab, the temperatures in the spring and early summer average 20 to about 26 degrees. We try to kill that down just a little bit to reduce stress on the fish. We can switch our systems to recirculating and add salt. We can use a little oxytetracycline that tends to work with one species of darter that has problems. The other thing we do is a lot of UV filtration, which slows the spread of kind of columnaris, which is our major bacterial disease. And if we can get the fish outside of their breeding season, that's preferable. We don't need any extra stress on our fish. We do all of our infections, but it's just typical bath methods. We use pretty standard 4,000 glycidia per liter. We can vary the number of glycidia we put in the bath based on the size of our fish. We just aerate really heavily. We keep them in there from anywhere from 15 to 30 minutes, depending on the size of the fish and the amount of glycidia. And then we're going to move them to AHAB tanks. And one of the reasons we like this method is we can kind of stagger these infections. And we'll have, if we bring back multiple females, we might do only one female at a time. That allows us to keep our broods separate if that's something we're interested in, and looking at different levels of genetic heterogeneity for our brood stock. And then also we can stagger our transformation. So a lot of our success is highly dependent on our state of our pond. So we do a lot of pond culture, and that allows us to kind of hedge our bets instead of the animals all coming off at one time. Maybe the pond's not doing well, maybe the animals aren't doing well, and you lose everything. It allows us to stagger those transformed juveniles over a period of a couple of months and sometimes. All our juveniles, all our host fish go into modified AHAB tanks, very similar to lots of other people. We use the really small tanks. We like the little 1.5 liter tanks. It allows us to isolate those fish, keep UV filtration in between them. Again, slowing the spread of things like columnaris, which is it's pretty rampant here. We have an interesting cold water strain of columnaris that was brought in back when the U.S. Fish and Wildlife operated our facility. It's not a warm water strain, which is very strange for us. And we'll chill those fish for about a week, and then that'll come up to room temperature, and usually we can get glycidia within 2 to 4 weeks. Here's our cups, very similar to everybody else. We're just gluing on Nitex screen. We either use 100 micron or 150 micron, depending on the size of the juveniles. And if we're holding fish for longer than a couple of weeks, we will feed them live blackworms to keep them in good condition. Here's an outside shot of our mussel pavilion. This is sort of a renovated pole barn that we added a lot of plumbing to. The windows are open. We don't have climate control, so it gets pretty warm. I mean, our temperatures in our systems can run up to 32, 33 degrees in the summer if it's really hot. We'll use chillers to kind of tamp that down a little bit, but we culture animals pretty warm. And again, it has two sources of well water, as well as water for our mussel culture pond, which we can filter to different levels. We can give it to them raw, or we can filter down to 100 or 200 microns. It's pretty coarse, but it seems to work for us. It's also power supply for pumps and things. And we're going old school with the original Barnhart mucket buckets. These are systems we're using the same buckets as we built these things in 2010. And they're just nested together with a head tank. Instead of using individual pumps, we have one pump driving 10 of those. And you can see that design there. We have 4 replicate systems, and we usually use about 5 cups per bucket. Here's the schematic from a long time ago. They're all upwelling. We don't use substrate for these. Again, based on Dr. Chris Barnhart's mucket buckets. We have 10 buckets per system, 5 cups per bucket, and we can stock these around 500 mussels per cup. So that gives you a capability of about 2,500 mussels per bucket and about 25,000 mussels per system. And again, these systems are unlike the original design, which was recirculating. Ours are flow through, so we're having a trickle of pond water going into the sump, which is about 1 liter per minute. We will hang a bag on that pond water to keep out things like ostracods and other crustaceans. And one of the big things we've been doing in recent years, if you seen any of our talks from, say, 10 years ago, we used to use a lot of aqua shade and tamp down the primary productivity of our pond. We thought that keeping it clear was a good thing, and it worked really well. We grew a lot of mussels. In later years, after we refilled our pond, we drained it to kill some corbicula. When we refilled it, we have some macrophytes that have been established, some of the Naeus species that are really common in our area, and they'll actually directly compete with algae. So what we have to do is actually treat the pond sometimes with herbicide. We use diquat dibromide to burn that macrophyte back. And when we do that, it frees up all those nutrients. It makes a ton of FPOM, and the mussels go nuts. So they love it when we treat the pond. And we can also, if it's cold, there's not a lot of macrophytes, we can fertilize with cottonseed meal. We also supplement our food just like everybody else. We have a lot of flow rate through our buckets. It's really fast, about 3 liters per minute. You can divide that by 5 to get the average flow rate for each cup. And we use nano and shellfish. It's dosed 6 times a day with a peristaltic pump every 4 hours. We refill our little beakers every day, so they're fed continuously. And we increase the amount of food based on how the babies are looking. Again, like some other people, I think it's really important to look at those mussels every single day. Look at their condition, see how they're doing. Are they active? Are they feeding? Are the guts looking good? That's an important thing. Here you can see some shots from this past year. Some of the Medianitis species we work with pretty commonly. You can see the little guys in the middle of some midge feces at about 10 days after they came off the fish. They're on 150. And then after about a month, they're already looking really good. They're up on 250 micron. They're about a millimeter long. So that's pretty nice. And we kind of maintain these cups we clean the cups out, spray them out about once or twice a week, depending on how dirty they're getting. We look at them every day, like I said before. And as they grow, we bump that size up because it really makes a difference as you want to push that water across those juveniles and have high water flow through the cup. So the faster we can get it on 250 or 500 micron, the better. 250 is usually pretty good, but 500 really nice. And after that, after they get 2 to 3 millimeters, to 2 to 4 millimeters, we'll go ahead and move those animals to our pond. And that can take anywhere from 2 months for some of the late transform species to about 5 months for some of the earlier slow growing species. Here you can see just a random shot. These animals are ready to go out into the pond. And here's a picture of our mussel culture dock. We actually have 2 of these now. We just finished or just almost completed a second dock, similar pond. It's on a 1.6 acre pond, pretty large compared to stuff other people are using. And but what it allows us to do, if we have to drain the pond for whatever reason, we can take that pond down and use the other pond, and we don't have to stop production for that. And we're using the what we call our subspecies buckets, which is again modified off of one of Dr. Barnhart's Flupsy systems. We sunk them down and powered them with air, and we're using the mesh in these is usually window screen, so it's about 1.5 mil, 2 mil, depending on which brand you get. We can also use Nitex, and for the past few years and previous years, we have made buckets with 150 micron mesh and with some success. But in recent years, we haven't had as good a luck with meshes under 500 mil. So if you're using a 500 mil mesh, your animals are at least 1 mil long, and you can, they seem to do a lot better. Here's just a picture of what the buckets look like. We clean these every week. It's a ton of fun. And we're stocking these. We're putting about 2,000 animals per bucket. We try to maintain that high density. When those animals are moving around, they keep things like bryozoans and sponges and other fouling organisms from encrusting on the screen. And we're cleaning them about every week, moving those mussels around, knocking off things like hydras and bryozoans from growing over the mussels. And we can hold these things we have held animals for years in some of these buckets. But usually, hopefully, everything's working well. We can hold them as little as in 2 months and get them to a stockable size. Here's a little quick illustration. I didn't do a fancy graph. It's just some pictures of our mussels from this past season. And these are Midinus parvulus, and I've shown this one a couple of times. This is a pretty rare animal. It's down to about 6 miles of river in the upper Conasauga River in southern Tennessee and North Georgia. It's one of our species we have to go out of state to get. So it's pretty rare. It used to be widely distributed throughout the Mobile Basin. But here's some animals, the left slide, and these are mixed with Cambarionio nebulosus. That's the yellow ones. The Midianitis are the kind of mahogany, chestnut colored animals, the darker animals. And this is about a week after they've been in the pond. And then 2 weeks after that, you can see the growth is very significant. This animal's growing rapidly. And we keep going. 3 weeks after that, they're even bigger still. And this is a picture of that 12th of November picture. They've stopped growing for the winter. Pond temps are down below 20. And that average size, those animals, that penny is 19 millimeters for scale, but the animals average with that batch about 13 millimeters long. That's an average size for the winter. Once we get them to a taggable size, which we usually determine to be 20 to 25 millimeters, so about an inch, we're going to tag with either the Loctite 410. That's the black super glue. We really like that. I like it mainly because it dries faster underwater than it dries in air, so you can keep your animals wet. It's good. We also attach either insect marking tags or typical shellfish tags with that glue, or just gel super glue. In the last couple of years, we've been using the pit tags like a lot of other people. We're using the 8 millimeter pit tags, and we're covering ours with, we're attaching them with gel super glue and covering them with plumber's putty. It seems to work pretty good. I've had some issues. If you always buy fresh putty, if your stuff's been open for a while, it can get a little brittle. If not, but it's not too expensive. And then to go on to stocking, we ask ourselves a few questions before we actually put animals out. We have the luxury of having a lot of state on channel bottom in Alabama, and that's usually where we're doing our stocking, but it's private land. The landowners need to be in on it. The Fish and Wildlife Service needs to be in and on it if you're doing a listed species. Make sure you're in the historical range. We don't do any extra liminal introductions. We usually want mussels to be present at the site, at least in some degree, even if they're just common species. And if we're stocking listed species, we would prefer there be another listed species on site, or it is critical habitat, because we don't want to introduce a regulatory burden on people if it's not already present. If we're not sure how the animals are going to grow, we'll do a silo test. And we also like to be cooperative with our Fish and Wildlife Service friends. We try to get the species leads out when we can, and we try to save them from staring at a computer too much. So we've got our local people out tagging mussels, scanning mussels. And we also try to monitor those releases. Again, I'll focus on these Midianitis because it's a fun species to work with. We're doing a new release. We try to monitor every year, but once we've had these reintroduction sites established for a while, we might go up to 5 years to monitor those and make sure they're doing okay. But we want to verify they're doing well before we kind of ignore them a little longer. Here's an example. These are some pit tagged animals going into the Little Cahaba River just down the road from us. And we saw about 30 to, we usually see with the pit tags, we've been seeing about 30 to 40% recovery. We're not digging all those animals up, but we're also not finding shells. So that's what we're seeing right now. And good growth. You can see the growth since they were stocked. Here's the next year. This is 2000. That previous picture was 2023. Here's the 2024 picture. And you can see both cohorts that have been stocked at this site. And that was not a big stocking. Those are only 100 animals to 200 animals. So we're getting pretty good. And we only tagged with pit tags about 30 animals from each cohort. Another thing nobody's mentioned yet is we tend to communicate best with our peers, but we also think outreach is pretty important. Here we have some interns from a local timber company. We're actually working on a mitigation bank, stocking on their mitigation bank, and talking. We have one of our Fish and Wildlife Service employees she's over at Auburn now, talking with some of those interns. And in the past, since 2015, we do a lot of hatchery tours to college age kids mostly, but also high schools as well. We've had 4,000, it's a little over 4,700 students go through our lab and talk about mussels. So sometimes they get pretty excited. If I got time, we'll talk a little bit about some success. And that, for me, success is not only seeing those animals you put out survive and grow, seeing their recruits, and that's pretty important to me. We've seen that with Toxilized Mussel for sure. We had some other cases where we may see recruits, we may not. It's hard to know, but these are ones I feel pretty good about. We've seen it in Toxilized Mussel multiple times, and that was only 4 years after stocking that we saw it again in 2023. So the other ones were Lampsals virescens. We finally getting recruits off that, as well as Hamiota pyrrhovalis. I'm going to switch gears a little bit, talk about a snail because it's kind of important. Well, kind of one of the reasons why we do this. The snail on the right is Leptoxus pictus, spotted rock snail. This animal we've been stocking in the Cahaba River since about 2010. We kind of stopped for a little while because we couldn't get brood stock anymore. And we couldn't get brood stock. We noticed these animals kind of disappeared from their habitat in the main stem, Alabama. And then that was very concerning because we just still don't know the reason why they're declining or have declined or been completely extirpated from most sites. But then we went back this year, and it's been 9 years since we stocked these animals, and they're thriving at this one shoal in the Cahaba. We don't know how far they spread yet, but now our stocking site is the best population range wide. So getting these animals out and getting them recruiting is pretty important. It can be in the near future pretty important survival of some of these species. So that's one I wanted to mention, even though it's a snail. Still fun though. But that's all I've got.
Well, thanks for everybody for tuning in to our talk. Just going to share some propagation techniques updates from the AWCC in Marion. And I'm Tim Lane, and I'm the Southwest Virginia Mussel Recovery Coordinator for DWR. And on the talk with me today will be Tiffany Leach. She's a mussel recovery biologist that mostly focuses on the propagation and rearing of mussels. And then our co-author, Sarah, mostly focuses on our field work and sampling and monitoring of animals that we're tracking post-release. And so this is our humble abode. It's not much to look at compared to some of the other facilities that have been sharing today. This originally was 3 trout raceways when Buller Hatchery was first built in the 1940s. These were trout raceways that ran off of a small spring that comes in off the mountain next to us and was diverted through. And by the late 70s and 80s were raising muskie using these sheds that they erected over these trout raceways because they built additional trout raceways down from us. And when a spill occurred in the 90s, Monte McGregor actually was like the state molluscologist at the time, or right before that. They decided to convert this space, the DWR, into a mussel holding or culture facility. And so Joe Ferraro, as many of you know, spent his whole career here kind of building this place up, keeping it up and making it function. And so you can see how mussels are distributed in Virginia. I think there's between like 70 and 80 species in our state. But the large majority of them are in the upper Tennessee River tributary portion on the southwest portion of the state. And within that little portion, you can see we're located on the extreme east side of that. If most of these rivers are in the valley and ridge type geomorphology, we're in the little section of it that's Blue Ridge. And so that means that the river that is flowing into us is not high in particulates and has pretty low hardness. It's a very good trout stream. And so that river, if you can see on the top of the map, there's flowing east to west through the Buller Hatchery. And our footprint are these 2 ponds in the buildings midway through the facility. And water is diverted to us from a low head dam which is about a quarter to a half mile upstream of us, that feeds water through a network of concrete pipes through the entire hatchery system. To each pond has a tee off of that main line, and we're able to fill water in from where the yellow circle is. And so water then flows into the ponds from west to east, and we have the option of diverting water into a head raceway. It's a concrete, like 6 foot deep raceway that extends from the edge of each pond all the way down towards those buildings at the bottom right there. And that's where we culture all of our mussels in the nursery stages and growing them, and then we take them back out to those ponds for culture. And so just to tell you about our ponds, where we have low hardness, a lot of the rivers where the broodstock originate are high hardness, like around 100. Our rivers, about 40. And so we do lime our ponds with pelleted lime. And we also fertilize our ponds with alfalfa and soybean meal. And we follow a prescription that's for fish hatcheries from the Fisheries Techniques book on that. So if you want to know that, reach out to me and I can give it to you. But we have 1/2 acre pond about 5 feet deep and 1/4 acre pond about 5 feet deep, about the size of an Olympic sized pool. And so I'm gonna hand this off to Tiffany, and she's gonna kind of walk you through a tour of the inside of the facility.
Okay, so first we have our RU3 system. We pull water straight from the head race. This system, RU3, is a flow through with an automated float switch. It's got 300 micron vessel filters going into a 500-gallon sump. What's really good about this system, especially where we hold our short-term brooders, is it provides our mussels with a natural temperature regime. So we've seen, year after year, our short-term brooders will spawn and release their conglutinates or their glochidia, multiple times a year. So it's been very, very beneficial for those guys. And just to add, we don't supplement any commercial algae to this system. So whatever algae is growing in our ponds, that's what our mussels are eating year round. So at the AWCC, when we're doing inoculations, we'll go to RU3 and grab our broodstock. We'll come into our lab space and we'll either extract glochidia with the syringe, or we might wait for our mussels to warm up in a beaker on the counter. Once the water warms up, they would start releasing their conglutinates. In some situations, we let our mussels, they actually release in the holding systems, and then we'll collect the larvae using a pipette. We have all these strategies. We're different years. We're propagating so many different mussel species. This past year, we produced 16 different mussel species. So we use all these strategies for that. So to do our inoculations, we mostly collect our fish using a backpack shocker or a seine. We do have a really great relationship with our fisheries biologists, and they sometimes collect fish for us, like our walleye. And then we have a partnership with Conservation Fisheries where they'll produce fish for us. Most of the time we use 3-liter or 9-liter AHAB containers with air for our infestations. You can see Haley in the middle. She's sucking up water in the baster, and just to kind of agitate the water to keep the glochidia from just laying on the bottom of the container. We typically do a salinity test to check for viability, and we also periodically check the percentage of closed glochidia during the infestation. And both of those help us to kind of figure out how long we need to do the infestation for. It depends on the number of fish you have, the number of glochidia, and the temperature of the water. These are one of our fish holding systems, our RPS. So we have 4 tanks with removable standpipes. We use those standpipes. We can pull the standpipe up and flush the system. So flush those juveniles from the bottom of those tanks into the plankton nets. That's where our juveniles are collected. The system has UV filtration and biobeads to help control the bacteria and the nitrogen in the system. We use these for our larger fish, like our walleye and catfish, and also our fish that like to school. In our middle building, we have 7 AHAB systems. These systems also have UV. They have 100 micron canister filters and charcoal canister filters. They have the bio beads in the bottom of those sumps. So we typically hold our sculpin and our different darter species in this system. So we rarely have any any fish disease breakouts, and we attribute that to our clean water source. So we start our production now in the winter. We've been doing this for the past couple of years. We usually stop about July. So the reason we start production so early, we have all these mussel species. We try to get our long-term brooders out of the way, where we are kind of limited on production space. So we start our long-term brooders in the winter, and so by late spring, early summer, we have space to start our short-term brooders. So once the fish are inoculated and go in our systems, after about 2 weeks, we start checking the bags every few days. And then once we see the live juveniles, check them every day. And then we move the juveniles into our RU1 or our ST system. So both of these systems, they're filtered through a 45 micron and a 5 micron bag filter. It goes into a sump that's heated, and then we use digital ball valves and Christmas tree timers to administer their flushes every morning. So you've seen these systems a couple times today. Our systems that I'm going to talk about, we've gotten our ideas from other facilities, from tours and talking to people. So this is our version of the sandbox system. It's pulse flow once a day in the morning. These are for our newly transformed juveniles up to 5 millimeters. These mussels are sampled every 7 to 14 days. And we do that because we want to control the presence of our flatworms and our chironomids. I've talked to other people who at their facilities and their systems, they're able to sample every 2 weeks to maybe a month. We just feel like we need to sample more often to control those predators. This is a closer look at the ST system. We use submersible pumps and the timers to pull in water from our sump, our larger sump, into the smaller white sump, and then the blue water supply tubes. That's what gives them the flush every morning. And you can see we use limestone sand as our substrate instead of like the silt that some facilities use. And we want to add more calcium to for our mussels, and it's and we can easily get it at a quarry near here and sieve it down. So this is our pan system. It's similar, so it's just like our sediment tank system. We took our flow through system and retrofit these pans, for this just as another sediment tank system. We use seed mats. Monte showed us how he uses his seed mats under his pans, under his tanks. So that's what we're doing in this system. So the sediment tank system, the sump, or the tank, the boxes are in a heated bath. And with this system, we couldn't recreate the bath. So we use the seed mats to heat our mussels. Our buildings aren't climate controlled. It's very drafty in there, especially this time of year. So we use the seed mats, and we also made a curtain system we keep it closed as much as possible to help keep our mussels warm. So our feeding regimen, we do 64 milliliters of shellfish diet and 32 milliliters of nano into 1 liter of water. So this is our juvenile food recipe that we use. And the picture in the middle, you can see the different food amounts. So we take that big mix and we pipette the different food amounts into each tank. So they get a breakfast, a lunch, and then a dinner. So for everyone for lunch, they get they get half a mil of our food mix every day. And we feel like where we do work with so many different species, we can have anywhere from 10 species to 20 different ones in these systems. And everyone, different ages and different sizes. So we just try to give everyone what they need. We also do a topsoil silt mix that sit below 100 microns. And we add calcium to that microwave silt to help give them extra food. You can see it in their guts. They're consuming this silt, and they seem to be growing well. So one of our newer systems is the plant box system, or our PBS. We took this design from Alabama and Virginia Tech. So we have refrigerated food mix. It's 20 milliliters of shellfish and 10 milliliters of nano to 5 liters of water. It uses a peristaltic pump to administer 5 seconds of food every 15 minutes. Sorry, 15 seconds of food every 15 minutes, and 24 hours, it takes all the food into the system. So with this system, we don't sample as often, every month or two. The mussels are bigger, so the mortality, is low. Also, the tanks are bigger. They're a little more cumbersome to carry around, so we like to do those not as often. And you can see our mussels, how well they're growing in this system.
So after they get through that size, that's really intended to kind of get them over, our mussels hit a wall at our facility around 2 or 3 millimeters. It sounds like that happens at the other facilities to some degree as well. But we're in cooler conditions. And so anyway, when they were getting there, we had that system Tiffany just described to get them like, a transition to going outdoors. And so they get a bigger water exchange with the pond. So when they are going out, they're used to getting quite a bit of pond water already. And so this is how our pond's set up. We have 2 jacuzzi pumps, one in each corner of our half-acre pond. And we do have the ability to use our quarter-acre pond for this, but we don't right now because it has so much turnover feeding the building. It never really blooms and sustains algal blooms, where the half-acre, we can cut it off, it warms up, and it'll bloom quite a bit. And so we just have these animal feeder pans that are lined with more coarse gravel, limestone gravel, and sand, and let them, this design really comes from Huadon and what she brought from growing pearls in China. And it by drilling holes through pipe, you get a spray basically, and it's feeding them. They're getting oxygenated, and the temperature's being maintained all without too much trouble. You just pump water through it. That's it. And we do have the ability we have a sand filter on the other corner with 2 vessel filters, but our pond gets so productive, sometimes we don't even use the filtration. It's easier to just let the really productive water flow over the animals, and then we'll just clean the pans more regularly. So that is a setback that we have to keep up with them all the time, but you can see how big some of the mussels we can get in really high densities, because the water can like when we do culture counter particle between 5 and 100 micron size for our pond in the peak of the summer, it can have like densities of like 6 million cells per milliliter. So I mean, they just have like unlimited food. And a couple other options. We have the upweller that you see on the left is just a simple design where the water comes into the tank. The only way out is through that middle standpipe, so that water has to come up through the screens. And then we have our Flupsy system, similar to what Michael described, but ours is floating instead of submerged, and the water is pumped out of the Rubbermaid troughs, and the 5-gallon buckets sit down at the base of that, and so you can see the water line is there at the top, and so water has to go up through the screens to exit the Flupsies. And we just use these for different species at different times. We like to have the options. Sometimes the pond can be like overly productive and we're not using this as much, and other times it's helpful. Just some recent production success. You can see we've got rough pigtoe, rabbit's foot, Appalachian monkey face, elk toe, purple warty back. So, a lot of the big mussel families, we're getting it done for Pleurbimini, Quadrillini, Anadontinae, and Lampsalines. And so I think, some of the rare mussels that we're working with, we don't have the numbers that some of the other labs do. but they're just a little more challenging to get them through the earlier stages up to that 3 and 4 millimeters. Once we get them there, our pond does really well for grow out for, like we've always wondered why. And recently we've been working with a phycologist, and guess what? He discovered a new diatom. It's new to science, that loves our pond. And so he's been he's trying to get this published right now, and it's probably going to be called Fragillaria holstonia, after the Holston River where we're located. But we know mussels like diatoms, but here's one that we didn't even know existed in there. It seems like that's maybe their main course in our pond, and it could be one of the reasons that we've had good success once we get them out on the pond. We don't have a long growing season. We have about from May through September, but they can triple and quadruple in size in just that short amount of time, and this is one of the things that they're eating. So just real quick, just to show you, we've produced over 7 million mussels at this facility. Of course, half of those were standing on the shoulders of many people who came here before us, talented and passionate folks: Joe Ferraro, Mike Pender, Megan Bradley, Nathan Eckert, and Amanda Wood, among others. 20 endangered species have been produced. And then, like you see the produce, but then what really went out into the river, it's about 175,000 grown out mussels, if we're not counting when they used to let things go at like 6 and 8 weeks early on. But 18 of those are endangered species. So pretty proud of all the work that's happened at this facility. And just want to say there's a link that my former supervisor Bill Kittrell's sister is a really talented multimedia person, and she did a tour of our lab. So it's right there on the right if you want to come back later and write that down. It's like a 30-minute where you can go through all the stuff Tiffany and I described, but like in more detail. And so just want to thank all our partners: Virginia Tech, Nature Conservancy, Tennessee, North Carolina, Alabama, Fish and Wildlife Service, and our interns from the last year, and other DWR staff. Thank you all for helping us be successful. So we can take questions.
I do have one. What calcium were y'all adding? And did you have the general amount? Were you finding any effects on your water quality with the calcium or anything like that?
It's reptile calcium. I think you would get it at a pet store.
Mm-hmm.
No specific amount. Whenever we sieve our silt down and we microwave it, in a beaker, and we just kind of take a scoop and mix it in with it. So it's not precise. We haven't seen any water quality issues.
Kind of like giving them a vitamin, we think. We're hoping that's kind of what it's like.
Have you seen positive growth? Like, are they bigger? Are their shells are stronger? Anything like that? I've been looking into it a lot with the snails. That's why I'm asking the questions, trying to figure out what I can use for them too.
We definitely used to see a really pale look to the mussels that we were growing here, both in and outside by the pond. And we think that since we've been liming and adding the calcium, that they've just had a much more natural periostrum and nacre kind of look to them, much darker and like filled in instead of like faded. And so, we don't have data to prove it, but we're seeing the results, we think.
That's perfect. Thanks. Yeah, we've very soft water here, so I'm kind of trying to figure out how we can deal with some of that. And I don't think we can really do too much to our pond the way we're located, but that's super. Thank you.
Any other questions? Yeah, I just have one. The quarry where you are getting your limestone, do they have a good amount that is in that, like, I don't know, 200 to 500 to maybe 800 range? Because what we've gotten some limestone down here from where Marion, North Carolina gets theirs, and we get a lot of like really coarse stuff, like 1 to 2 millimeters, and then we get a lot of like crushed, almost powder. It's hard to get the kind of in between. Y'all get that kind of in between?
Ours, I would say ours seems to be about half is above 500, but half is below. And so we use a sieve shaker.
Yeah.
I don't know if we showed it to you when you visited, Chris, but it's just a mechanical sieve shaker, and it will stratify all that different sizes for you really efficiently. And so the majority of what we use is about like 100 micron. It'll fit through or through a 100 micron, and then the next is technically called 125, but like fits through a 150, I think, micron sieve, and then we'll step them up gradually, but we use mostly things that are less than 200 micron for the majority of the culture till they go outside.
Yeah, I forgot to mention that we use different sizes just based on their size and kind of graduate. It goes to a 100, a 200, a 3, and a 4, and then on from there. 5.
Last call for questions for Marion, Virginia. We got time for one more.
Nobody else has one more question. How did y'all get hooked up with the phycologist that found your new diatom?
Interesting enough, he reached out to me asking if there was anything that we'd ever done on diets of mussels. His name is Dr. Bruce Cahoon. He's at UVA Wise, and he's kind of an expert on diatoms in streams, and so he started helping us just sample our pond, and then we got a recent paper you can look for in Nature on what are mussels eating, and we did an eDNA study with them, and turns out like you can use eDNA and figure out what they're eating from the water samples. And they definitely are highly focused, as you might imagine, on algae, but also on fungal spores seems to be really important. And, we hear like bacteria is important, but it seems that they prefer not to eat bacteria unless it's like a last resort. So anyway, we're always learning, but yeah, it was just random that he reached out to us. So I would recommend reaching out to your local phycologist. It's helping us learn.
Thanks. Do you have that video?
Yeah, I'll try to find a link and put it in the chat.
Yeah, thank you so much.
Mhm.
Okay, well, I'm Sierra Benfield. I'm the coordinator here at the hatchery. I have Kelsey here with me, Mansell, and she's our technician. We're our only 2 full-time staff. We do have 1 temporary, and she's out scrubbing some snails. But we'll kind of go over a little bit of a tour. But first, I wanted to sort of go over like if I can get it. There we go. Our facility goals, I think all of the facilities kind of have similar goals, but I wanted to highlight that ours in particular are focused on informing management strategies for the agency biologists. So our agency actually places us under Sportfish, like a Sportfish hatchery. So our upper management and chief staff only really look for production. Their expectation is a little less focused on research than some of the other facilities. That's shifted a little bit since our previous coordinator, Rachel Hoke, has moved up to Raleigh. But we still are a little bit limited in what we're expected to do research-wise. But we are still able to focus on some things like host trials and especially production strategies that we're kind of allowed to do. And then anything outside of that has to either be especially requested by one of our field biologists or we need a student to come in and do. So we do try to support graduate students 1 every year or 2 if we're able to try and get more of a research focus going on in our lab. We also have the opportunity to do a lot of life history studies. North Carolina has a ton of endemic species, just like everybody else. But one thing we've started to realize now that we have a agency geneticist is that a lot of our weird mussels are actually new species. So we've been doing a lot of work with species descriptions and that kind of thing. But to kind of back up, Kelsey and I both started in late spring of 2022 in our new facility. So the hatchery, the new hatchery here came under construction in 2016, and Rachel started trying to do production in the new facility in 2018 and really 2019 with just a few systems built out enough to run, but still not even fully built out, the ones that she was using, and was trying to build out as she went. So when we started, the hatchery only had 7 systems completed out of 26. So in 2022, we were able to really bust tail and finish the build out on that. And then the entire new facility was plumbed and built and put together by our staff and temporaries from other hatcheries too in the North Carolina hatchery system. So we're very proud of it, even though some of it is definitely crooked. It does work. So we started out in this new hatchery, neither of us having any aquaculture experience. And then 3 months later, Rachel was promoted to assistant chief and left. So we had this big facility and no clue how to run it. And the new facility had a lot of issues that Rachel was still trying to figure out. So when production started there in 2018, 2019, every lot in the new building kept going to zero, even the not very sensitive things. So we kind of had a lot of figuring out to do. So when we started, we did 1 million tests. We had 26 systems, and by gosh, we used all of them. We tried everything under the sun in terms of holding systems, substrates. We have 3 different water supplies, which we'll get into. We tried different calcium treatments and different filtration regimes, just trying to find a way to keep the mussels alive to start with and then go from there. And we learned a lot. So from June of 2022, right as Rachel was leaving, that's what our mussels looked like. These are the same species. These are both Carolina Creek shell. So the 2022 lot, and then we made a lot of changes, learned a lot, and then 2023 lot. And then these are the same. This is the same mussel, Carolina Creek shell, from this year's lot compared to last year's, the same magnification for all of those pictures. So we've really come a long way in our own understanding of how to run the building, but also in developing methods for the new hatchery. So to go over our water supplies, we're kind of unique in that we have 3 separate water supplies, but they do all interact with each other in kind of crazy ways. We have what we call our warm pond. It is mainly surface water, but it is fed by the cold pond and it is flow through. So it's kind of difficult to maintain a good algae bloom in, though we do try. We'll have catfish in there and fertilize it. We also have to drain it every 2 years because it flows directly into a creek that flows directly into Lake James. So we do that drain not only to fertilize it with like pelleted lime, but also to prevent escapement since we do have mussels in subsies out there. So that's the warm. Cold, we kind of have 2 different supplies that we refer to as our cold, but they're very similar. Our cold pond and our creek intake, you can see on the bottom there, are both almost entirely groundwater. So the nice thing about those is that they're, while they're not very productive, they are very temperature stable. So they can help us to mediate the temperatures in either building, especially the mussel shed. But the cold water from either intake is used for trout rearing, which we'll come back to. And then we also have a well, and that well is also used a lot for trout rearing to supplement the cold water. And it is hyperoxygenated with an oxygen machine. And we have the well running to all of our faucets and hoses, so all of our outlets that we can use are mussel and fish safe. But the funky thing that happens with all of this that I don't think was really foreseen whenever the new building was built, when our new hatchery was built, the trout hatchery was also upgraded and capacity dramatically increased for the trout side as well. And since the cold pond supplies a lot of the water to the warm pond, when trout production increases, the turnover in the warm pond plummets. which means our pond that used to top out in the summer around 28 to 30 degrees now tops out in the summer around 37 degrees. So we are no longer able to rely very heavily on our pond for grow out of our mussels because it just toasts them. But one cool thing that we have, since we have those three supplies, is every single system in our new hatchery, all 26, have access to all three water supplies, and we can mix them however we choose to. And we can automate those water changes with that little blue valve, or we can do manual water changes. For filtration, we have a filtration building that goes to our new hatchery facility, and the two sides run in tandem. One is warm and one is cold. And the water flows through drum filters to a holding sump. And then as we increase demand in the hatchery building, the pumps send it through the UV in sequence to the building. One caveat with this is that the filtration building is about 100 yards from the water supplies themselves. Twice that for the cold, but then it's about 600 yards to 800 yards from our hatchery building. So there's a lot of time underground sitting in a dark pipe between the filtration building and the hatchery building itself. And so when we were doing some testing on our water, I actually worked with a microbiology professor at one of our local universities and found that our incoming warm water, which is what we would normally use for all of the juvenile mussels, what Rachel had been using for all the juvenile mussels, by the time it got to the hatchery was basically sickness soup. So what survives the UV is like encysted gram-negative bacteria, fungal spores, flatworm eggs, and all of those things were then proliferating in their time sitting in that dark pipe, and then being dumped into our mussel systems at every water change. So one change that we made after doing all of that testing initially was turning off the UVs at our intake filtration in the filtration building for the warm side, and instead UV filtering on the systems themselves. And we did not have as many problems with disease anymore. For a lab, we actually have a really, really small lab, but it has all the things that we need. The pictures kind of make it look bigger than it is because there's a wide angle. But we do have microscopes with cameras, just like everybody. We have a cell counter, which we use all the time. We have sort of a hood, but that's mainly used by sport fish when they do otoliths, since they share our lab. We have a small autoclave, and we also have a laser engraver, like folks were talking about earlier. We don't use our laser engraver a ton because they used to use it a lot back in the day and started seeing a lot of erosion on the mussel shells. And we kind of think speculatively that some of that is just because a lot of those sites have become more and more unstable over time. And so they're shifting around, getting sandblasted. But we're hoping to start using it more and more as we find better sites. For our infestations, we have been working on developing protocols by species because we use a very wide range of hosts, anything from margin mad toms to largemouth bass and lots of things in between. And they have very different tolerances in terms of like infestation time and load and that kind of thing. So we've really been trying to narrow that down by species, but we predominantly do McDonald jar bats, and we will kind of stagger when they're removed to try and hedge our bets. So we don't kill the fish, but we attach as many as possible, that kind of thing. One really cool thing that we were able to do this year was testing different extraction methods. For two elasmodontas, we did Appalachian Octo and brook floater, testing serotonin exposure versus needle extraction with 3 gravid females in each treatment for each species. So for our treatments, we had what we called initial release for the serotonin bath. So those were the glycidia that the female released during the serotonin exposure. And then the additional release after water change is after we did the 100% water change, we let the brood mussel continue to release until she was done, and then did our infestation another round with that. And then we had our normal needle extraction. And what we found was that with both species, that initial release into the serotonin, so the glycidia that were actually exposed to the serotonin bath as well, had a significantly lower transformation rate than any of the other treatments. And that was for both species. But for Appalachian elk toe, the additional release, so the ones that were after the serotonin exposure, had a significantly higher transformation rate than any of the other treatments. But in the brook floater, the needle had a significantly higher transformation rate than any of the others. In fact, it actually had 3 times higher transformation than either of the serotonin treatments. So this is kind of a cool way that we're able to incorporate it into our normal production, but still kind of help narrow down some of those protocols. So really cool. For our host systems, we do have some long-term holding for hosts. So we receive white shiners from Edenton National Fish Hatchery, and we use those for several species. So they're sort of on a rotation. We'll use them for one species for a couple of rounds, and then we'll switch species and try to maximize how many times we can use those shiners before we need to swap out. So there's a little rotation system there. All of our incoming hosts go through a intake protocol in quarantine and do formalin treatments. We normally do a low dose of formalin for 2 to 3 hours, depending on the species, and most things tolerate that really well. And then all of our systems are very large, but they are designed for host trials. We have 24 tanks per system. Each system is completely isolated, and the tanks can be isolated with the catch cups. So the idea with the 24 tanks is that we could do 8 host species with 3 replicate tanks randomly distributed throughout the system. One other really cool advantage to having such large host systems and having so much gallonage is that they also double after infestation season as a way for us to provide grow out space for Conservation Fisheries Inc. Whenever they propagate North Carolina species. We can decon our host systems and flip them, and they actually serve as pretty decent grow out systems for those fish. So dual purpose. We also recently have started placing socks at the base of our gutter systems. We were finding that we were having lower juvenile counts than we were expecting based on our attachment, and realized that we were having some escapement, especially when the catch cups would overflow over the weekend if somebody wasn't there to rinse them often enough during the big drop. And that has helped a lot. And then you'll see in one of the pictures, the ugly looking black covers on the host system. Those are guards for some of our fish. One of our host system quirks is that some of the baffles have big gaps under them, and we filed them down and they've gotten better over time, but some fish do still escape and can jump out of the gutters. So for those fish that like to escape, we put gutter guards on them, and that helps a lot with that issue. We also have one system that's more similar to some of what we've seen earlier today with the round tanks designed for larger hosts that like to school, that kind of thing. And it works really well for things like bass. For juvenile care, we start them on compost, very similar to what Tim was talking about. And we also add the reptile calcium to our compost. I don't think I mentioned it, but our incoming water has a calcium hardness of 20 at best. So we do all sorts of things to try and increase that. So we start them on compost with reptile calcium mixed in. And then as soon as they get a little bit of size to them, usually within a week or 2, we start adding in fine limestone sand. But we do weekly sediment changes, and our sediment changes are pretty aggressive cleaning wise. And after the sed change, we dose with probiotics like the clear flow that Monte mentioned, and we also use nitrifying bacteria. And then we maintain a biofilter culture and replace biofilter bags in those systems after the sediment change as well. All the mussels are scored weekly for their overall body condition and also the food in their gut and fecal line. And we do, this is a relatively recent thing we started doing, is preventative fluconazole treatments for fungus-prone species. So we kind of have 2 or 3 lots every year that seem to have a big issue with this, right around a month old. So around 3 weeks, we will dissolve fluconazole tablets into the substrate of those species in particular. And we have seen much less trouble with water molds and fungal diseases since we started doing that. We also have started making sure that every production lot goes into at least 2 separate systems. So we have 26 of them. A lot of them, the rows can be isolated. So we'll do, the same basin in one system, but we may have different species in the rows with socks on them, that kind of thing. But that way we sort of hedge our bets. So since these systems were new, especially when we started, there were a lot of quirks and things still being worked out, and we would have system failures. So when we first started in 2022, if all of our Appalachian Elktoe are in system M and then M failed, there went all of our Appalachian Elktoe. So this has really helped us make sure that that doesn't happen again. And we also split our mussels to lower densities at 1 millimeter and at 2 millimeters. And all of our systems, the tanks and troughs, can run with 1 tank or with all 24, and it doesn't place any strain on the system. So for tanks, like I said, they're expandable. We can start with 1 tank, and then as the mussels grow, add tanks to it to split them. And that's very easy for us to do. And it's nice early on when you're still doing infestations, counting juveniles, you have fewer tanks to care for, and that expands almost as the other work kind of finishes up. So it works out nicely. You'll kind of see in that middle picture for the big tank systems, the water is dirty because we just did a sediment change and it's composty. But we'll start the really small juveniles in little tubs on top of the tank. And then after a couple of weeks, we can drop them down into the bigger tank. And we've seen some better survival with that. But yeah, the main tricky thing with these is just that they're very large tanks. Each tank is 8 gallons. So they can be kind of cumbersome for working with, but they do really well with fast growing species. For the trough systems, they're easier to care for because they're smaller holding units, but we do have lower capacity in those. We found in these that the slower growers, the more delicate things tend to do better. That might be a function of just that it's easier for us to care for them. But they do work well for that. The only real issue with these is there's not a good way to isolate holding units. So if they escape out of the tub into the trough, which is a water bath to maintain temperature, then they're just kind of out there. We'll catch them in the sock when we do a sediment change, but there's no way to isolate lots unless they're on different levels. So you do see we have 2 levels in each trough system, but we can only do 1 lot per level. These do seem to be less prone to disease outbreaks, but I think it's because they're easier to clean. But yeah, another cool thing with these and all of our systems is that all of the parts are exchangeable. So we can swap out the spray bar for an entirely different configuration at the drop of a hat if we decide we want to do something different. For our feeds, we maintain target concentrations in the water volume. And we test that usually Monday, Wednesday, Friday. And then once the season kind of stabilizes for our pond and everything, we can kind of back off on how often we're using the cell counter. But we use a slightly different mix of feeds depending on the system and how the graph looks on the cell counter. So we have kind of a standard target, and we can tell by looking at it if there's a dip in a certain cell size range, which of the 3 feeds we need to increase, because they all have slightly different average cell sizes. But we do the standard nano shellfish TP. And then we also will dose our systems with probiotics. So the nitrifying bacteria, just the standard culture. And then we also make a mix of the clear flow that we add to our systems. In our mussel shed, the old mussel shed, it's still our grow out space. It's still our predominant grow out space. It also has all of our arcs and broodstock holding. So a lot of our short-term breeders we keep in there, and they spawn and release in those tubs, and we collect them for infestations. And then we also end up kind of taking in refugees. So if a dam removal is impending, we may get a call from biologists asking us to hold some for a season until things settle out or things like that. Or following flooding, like with the hurricane, or in trying to prevent things from being lost to flooding. So we use this space a lot, even though it's not the main production space anymore, to the point that we've actually had to expand it a good bit and build grow out space into the old production space. We also use it for grow out. We used to do most of the grow out in the subseas in the pond. And in the last few years, since trout production has ramped up, we have not been able to do that. Especially our mountain species, most of them start gaping and dying around 25 degrees C. So they couldn't tolerate the pond before it was hitting like body temp, much less now. But some of our things can still go out there. One interesting thing that we found, so when we first started, we were having a lot of mortality in the subseas from caddisflies. And they initially thought that the caddisflies were making cases out of the mussels, but they were actually net spinners. And I guess the mussels were the only suitable substrate in there. So we added aquarium plants to our subseas. It was a great intern craft activity, selling them in there with fishing line. And the caddisflies spin their nets on the aquarium plants, and the mussels have been much better off for it. So that's a funny little quirk. But even though the pond gets too hot, because we have the extra water supplies, we can mix cold water into the mussel shed to regulate the shed temperature to that 24 sweet spot. I'll just have a little slide for the snails, mostly for Loretta. But we do maintain an arc of magnificent ram's horn, and then we do production as requested by field biologists. They have sort of a similar setup to a lot of the mussels, recirculating with lots of filtration. And going back to the calcium, we dust their food with the calcium powder, and that seems to do well for them. For 2025, we have lots on the docket. The italicized ones are host trials. That funny one at the bottom, Microdonta obscura, is actually a species description that our field staff have been working on for a long, long time. And it is a new genus and species, which is pretty cool. And we'll be trying to do production of those this year as well. But with that, I can take any questions.
Yes. Nice picture. Thanks, Sierra. Any questions? We got time for one or two.
Hi, Sierra. This is Monte. I had a question about use of your serotonin, and it seemed like you got some mixed results with that. So I was really curious about, after the packets are spit out of in your serotonin solution, do you like rinse them for a period of time before you use them on the fish, or is there any time period allowed to that serotonin to kind of get off of the larvae? I'm just curious about that.
Yeah, so we Whenever we siphoned out the water from the serotonin treatment, we siphoned it through a screen. So the Glycidia went onto the screen and then were rinsed into clean water after that. That was from the same system. So we didn't shock them. But yeah, they were removed from the serotonin bath at that point and rinsed into clean water. And then we went straight into doing the normal infestation protocol. So like the salt test and counting, and then right onto the fish. So there wasn't a huge time delay, but they were removed from the serotonin at the same time the brood mussel was. Does that help?
Yeah, that answers my question.
Hey Sierra, this is Tim. I wanted to ask about the fluconazole. Do you have to get like a vet to prescribe that to you, or can you like, buy it at the pet shop or what?
We do get it from a vet. So our hatchery supervisor that's over all of the sport fish hatcheries has a vet contact that is like contracted through the agency for any prescriptions like that. And we get prescription fluconazole from that vet. And it comes in tablets, and we'll dissolve the tabs, and they actually settle out. Even once they're dissolved, they'll settle out over a period of time. So they'll stay in the substrate. So we just mix it in with the substrate when we add it, and it does pretty well.
Yeah, back to that, to the serotonin question. I was curious, what would you do moving forward, like for this year? What did you notice in terms of like harvest of the broods? And those species are kind of mucus producing
Yeah.
species. Are you going to just go with the needle? What do you think you're going to do going forward?
So the thing that we were interested in, especially with the serotonin, was for some of the Appalachian elk toe. Our little Tennessee elk toe tend to be really small, even as adults. Whereas the brook floaters, which actually did better, the needle extracted group did do better in the brook floaters. They're big, and they tend to tolerate the needle extraction pretty well. So for those, we'll probably stick to needle and then use the serotonin for things like the elk toe that are so small. Same with like slipper shells. They're just little and don't seem to tolerate the handling stress as well. And we'll just go by species.
Nice. Thank you. We got time for one more? All right. Well, thanks, Sierra. And I guess that leaves me. So what I'm going to do is I'm going to let my two co-workers introduce themselves here on video, and then I'll start sharing my screen.
Cool. Well, I'll go ahead and start. Hey, everybody, I'm Loretta Lutakis. I work here at the Yates Aquatic Conservation Center with Chris Eads and Craig Lawson through North Carolina State University. I really focus, kind of do everything, but I really focus on the magnificent ranch horned snail. So thanks for this shout out, Sierra, and the in vitro lab. So then I'll toss it over to Craig.
Everybody, I'm Craig Lawson. Hopefully you can hear me. The mics are kind of weird in this room. I've been working on the AV things over here. I'm sort of the newest member of the team here, working with Chris and Loretta at Yates Mill. I'm a generalized aquaculture guy, specializing in researching and closing the loop on new species. I do most of the fish work around here. I'm fairly new to freshwater mussels, so I'm learning a lot about their life cycle and their culture here. So it's been really great to hear how everybody does things differently in facilities. Very exciting. So thanks everybody.
All right. All right. So I'm going to just give you a little bit of a tour, but focus mostly in on how we're doing just our mussel rearing. Um, just a little bit about us. We are located in the Neuse River Basin, just south of Raleigh, North Carolina. We are on a historic mill pond. That mill you see there in the picture dates back to the 1750s. The land around it operates as a county park, and our facility is actually attached to the county park museum. We are an existing research wing within NC State University, so we're operating with the county as a partner. Our facility, I've been in here at NC State since 2001. Doing mussel research, but this facility was established in 2023 as a result of a settlement over a highway project, a large highway project around Raleigh. That highway project went through habitat of 3 federally listed mussels that you see there, as well as the endangered Carolina mad tom and the Neuse River water dog. A little bit about our water supply is it's a 20-acre mill pond. You can see our watershed there. It's kind of a mix of a little bit of urban, a little bit of university-owned ag land, and a little bit of forest. So it is fairly highly productive, but we are subject to high turbidity after heavy rains. If we get a couple of inches of rain, our pond can turn a little bit of brown. So here's kind of the layout of the facility. We have about 4,400 square feet of indoor and outdoor space, and I'll take you through that room by room. This is our in vitro lab, where Loretta spends a lot of her time. Office where the 3 of us share. Our classroom has been great. We will often start off tours that come through. We have outreach, being connected to the park. Outreach is a really natural part of what we do. So we'll bring in classes or things like that, show them all the cool mussel videos and get them excited before we go through. Going down to the other end of the building is where we'll start this tour. This is our fish quarantine. We have insulated tanks with glass windows. We're using ultra-filtered pond water, and we have 3 systems in there that are pretty good for quarantining fish. Our microscopy workspace, and in that space, I really love this. We've repurposed this necropsy table and an old living stream that we're using to heat water for sediment changes. So we have 2 sprayers for different water temperatures. This is another thing that's been really great in our outreach tours is this filtration display. That picture there on the right, you see you actually have 2 streams flowing from the top shelf down to the bottom, and this is flow-through pond water. And that's flowing through constantly, about 1 to 2 gallons a minute. And so people are able to come in and see real time, even with flowing water through a tank, mussels are, visibly cleaning our water for us. So we like showing that off. That's next to our juvenile mussel production. We have 3 AHABs and a custom system, which I'll show that here. And so this is something that we designed, had conical bottom tanks made there by Hydro Composites. And these catch baskets are baskets that we made as well. We took 6-inch PVC pipe and cut it out, and then used a plastic welder to weld in stainless steel mesh. And this system has been really great for shiners. We do a lot of short-term brooders here that use Luxilla shiners as their hosts. And so when we're doing lots of small batches like that, we get a brood released at a time. We can inoculate 10 to 20 fish and put those in 1 tank. And it's worked really well so far. Snails. This is the endemic. It's a fairly endangered North Carolina endemic that we're raising along with Sierra and Marion. Craig is getting into a little bit of rare fish propagation and juvenile mussel rearing, which I'll get into. And this is all outdoor space where we're doing our final grow out and some broodstock holding as well. All right, so let's go into where our water comes into the facility. So here we are looking at the intake. That intake goes through 2 mm screen, and we have these dual self-cleaning intake screens with rotating spray bars that keep it all clean. So this intake it requires very little maintenance. I think maybe once or twice over the last year, I went off with a scrub brush and sprayed it down. So that was really nice. Our pump house is a separate facility. It holds all of our pumping and initial mechanical filtration. Our pumps, everything is in duplicate. So in the event of failure, we don't lose water. We can pump up to about 400 gallons per minute if we want that much. This past year, we've operated on about 80 to 100. And that's our first year of operation. We've operated on about 80 to 100 gallons per minute most of the time. I expect that to go up this year with more mussels. Our incoming filtration goes through these. These filters are made by Evoqua. They are stainless steel screen, and they are auto backwashing, means that at whatever interval they come on and those nozzles suck the screens clean. Water is normally flowing from the inside out, and so those nozzles vacuum the screen and wash that back down the drain to the pond. And then our outgoing filtration is similar, also filtered to 25 microns and then UV treated before it goes back to the pond. What's really neat is we're able to look at the pressure within those filters online, and this is kind of how it works. So we got 2 pumps that go through 4 75 micron filters in parallel, and then 2 larger 25 micron filters in parallel, and then that's on to the building. That auto backwash is great, but we still get bryozoan growing in there. Especially during the warmer part of the year, we get manganese scale that clogs the filters, and neither of those the backwash will clean. And of course, filamentous algae can tend to push through the screen and eventually accumulate in culture tanks over time. So we've been having to break these filters down, clean them about every 1 to 4 weeks or so, more often when the warmer weather, less often in the cooler weather. We are getting to the point where we think that our water is so productive that filtering out that much water it's tough on the filters we have. It's a lot of breaking it down, taking it apart, cleaning it, putting it back together, and they're starting to get some wear and tear on them. So we may end up backing off on not going to 25 microns on all of it, and just doing some of that inside in smaller batches for the younger mussels. So this is what our early grow out looks like. This is where mussels go straight off to fish. This is a 5 foot by 2 foot aluminum shelf from Granger. The tanks are 2.5 gallon fiberglass. They come from US Plastics. So in 2 racks, we've got 42 total tanks. And so our head tanks fill, and those happen. They are actually controlled by irrigation valves and an irrigation controller by Rainbird. These are great. They're highly programmable. We can do up to 4 fills of the head tanks per day. We choose the time, and it controls. That controller will control 6 groups of valves. They're really affordable. But the only caveat is that the water has to be under a pressure really greater than 30 psi. Our water coming into the building from the pump house is around 50 psi. And again, that's the 25 micron filtered pond water that's coming into those head tanks. They're 25 gallon head tanks. Within the head tank, we have aeration. There's a UV sterilizing light. We have a programmable heater. So the water coming in gets heated up before it gets used. And then when they drain, that is controlled by a separate solenoid. We get ours. This is the stainless steel. We get them from electricsolenoidvalves.com. And we're using this programmable timer. This is a yard state timer, very affordable from Amazon. And they've worked fairly well, and they're programmable up to 7 times per day that you can use those to turn over. All right, culture tanks. They're 2.5 gallon, and we're doing right now 2 turnovers a day. They have aeration. We're using this heating mats as well, greenhouse heating mats, and a thermostat. And these heating mats, we have 1 heating mat for the whole shelf. That's why it's $250 each. I think you can get them a little cheaper somewhere else, or a similar product cheaper elsewhere, but these have been great for, we heat it up to 24 to 26 degrees C. We have individual acrylic lids to keep chironomids out, or to try to keep them out. And then we have inside the tank these screen inserts to try to prevent mussel escape. For animal care, we're just batch feeding, like many of y'all, twice a day, using the typical reed products. And then this is how we're making our sediment. This is something that we have found to be at least reproducible, not having to go out and collect. We are taking the worm castings, drying it out, sieving it out below 100 microns, and then also using some of the limestone less than 100 microns and mixing those two. And it seems to work pretty well. It gives us a substrate that has organic content, but it also has the calcium and buffering capacity of the limestone. Our sediment is changed weekly. For cleaning, we found that Instapots are really good for substitution for like an autoclave for sterilizing your sediment. And then we will aliquot the sediment into those Tupperware containers and freeze those for use the next time, so our sediment can be reused. So this is kind of our first year results from that first stage of grow out. And so this is you're looking at survival there for the whole grow out year. So that includes some of the next phase grow out, which I'll get into. But we're fairly happy with these survival results. Mussels are reaching 2 millimeters around the 2-month mark, between 7 and 9 weeks. We'll have a batch or two here or there where survival has been less. And again, those survival numbers you're looking at there, those are all fish produced survival numbers. So in vitro overall survival is going to be less, but so yeah. So from there, like all of y'all, we're finding that the 2 to 3 millimeter mark is the time where they're outgrowing those boxes and they need to move. And they're moving into this system here. And this system is designed to be as flexible as possible. So they can be completely flow through, or they can be recirculating, or any combination thereof. We're designing it for flexibility. So if we want to hold broodstock in there, we can. The recirculation has been uses automated water changes with conditioned water. And so we made these actually using food racks. These are racks used in the restaurant industry to hold these food grade plastic lugs is what they call them. The rack itself is about $900 right now. You can get aluminum ones for about half that. The tanks themselves are about $20. This is the reservoir that we're using for our water changes. It's just an old living stream. It fills with the irrigation valves that I showed you before. It can be heated or cooled depending on time of year. And we've added this stainless steel tabletop, which creates a nice workspace. And so when we do water changes, right now, the juveniles I have in there, they were produced this year. We're doing a 25% change 4 times a day. So what happens is a solenoid on the side of the tank opens up, drains about 3/4 of the tank. And then after that, the reservoir comes on, pumps water over, and refills the sump. And then at that point, the reservoir refills with 25 micron pond water, and that water is then heated. This is how we're feeding. Inspired by Tim and I'm sure others. We have this refrigerator that sits on top with a peristaltic pump. We're using this Liter Meter 3. This is it's a nice tool for, it's easy to calibrate. The head takes a little bit of maintenance, but I really like it. It runs auxiliary pumps as well. So I can run those 3 systems just off of this one unit with 2 auxiliary pumps. The culture tanks, this is how they're designed. So everything, water goes from front and is delivered to the back. The standpipe is in the front. So all the controls are in the front so you can access it. And so the barb, we're using barb and hoses for ease of access. And these tanks are packed in order to get as much, as many mussels, as many tanks in the small area that we have. But it's hard to look in a tank, when they are all together, but you can access the tank fairly quickly by just unhooking the hoses, and you can have a tank out so you can see it in about 15 to 20 seconds. And then from there, our final grow out is outdoors. And so this is actually a bit of a screened in porch. And so those deep blue tanks on the right, this is all flow through pond water. That's where we're holding fish. And there on the left, those are these racks are they're designed for flexibility as well. So they are 8 feet long, 3 feet wide. I got those from Pentair, and they are also the 25 micron pond water just flowing through. There on the screen-in porch, we have the vinyl curtains that allows us to do some partial heating in the winter, just to make it a little more comfortable for us. If you're sticking your hands in the water, you're not dying out there. The water's not freezing. And then these troughs are nice because they can accommodate tanks. They can accommodate these modified milk crates. We've taken milk crates and welded on mesh of various sizes. And then water is delivered by this hose into a slitted standpipe of sorts, so that we ensure flow from the inside out, and all the mussels within a cage are doing well. So these are our results from year 1. So using a combination of all of those things, starting them in the 2.5 gallon tanks, moving them to flow through at about 2 or 3 millimeters. And then when they're outgrowing those stacked boxes in the food racks, they can come outside. And so by the fall, in one growing season, this is what we've got. Our Varicosa are 15 to 28 millimeters. We've done Lampsilus cariosa, roughly about the same size, doing really well. And Strophitis undulatus also getting nice and big. So we were pretty happy to see that. And let's see. So yeah, as I said, best success going to flow through at 2 to 3 millimeters. So that is all I was going to present to you guys. All right, welcome back for everybody that is here. I wanted to start us off with a question I had, kind of triggered by Jess's presentation, but I wanted to pose it to the group. I don't know, Jess, are you still here?
Yes. Hey, Chris.
Okay. So yeah, Jess, I was noticing when you were talking about the numbers of mussels that you were producing, you talked about a Lampsilus, and I don't know, you said you had, produced 150 to 200,000 mussels of some Lampsilus. And, kind of, it looked like you, like us, are starting juveniles off in these kind of like 2-gallon containers. And we don't want to overcrowd these containers. So I was curious how and I want to put it to the whole group, is how are your hatcheries, how are you dealing with these just species that are large volume where you're getting hundreds of thousands of mussels? If you're dealing with systems that are, starting them off in fairly small systems, I don't want to clog up all of our all of our labor on some of these more common species, but I don't want to spend all of our time on them. But so how do you deal with that many? And does that make sense? Without it, taking up all of your tanks or all of your time. I'll put that to Jess and to anybody else. I know White's or Harrison Lake deals with some pretty large batches of mussels too.
So, Chris, I mean, one thing for the projects that we're working on, just using, like, say, the South River project as an example, you know, they're NERDAR projects, they're natural resource damage assessment and restoration projects. And so there's goals in terms of trying to return, lost mussel service years in those projects. It's kind of just built in inherently to the injury assessment all the way to the valuation and then ultimately what is restored. So there's some obligation, on the front end planning for those projects to get some big numbers out, to try to recoup, the lost mussel service years. So that's one of the reasons we are kind of committed to some of the species that we can produce bigger numbers on. But to your question in terms of how to handle it in the lab, we have say in the main mussel lab where we're doing the Atlantic slope work, which actually it's exactly the same in terms of the other lab, but we have what is it, 160, we have capacity for 160 boxes of those little 3-liter AHAB boxes, right? And so each of those AHAB boxes, the most that we'll put in there is about 1,500 mussels. So, we're looking at, a couple hundred, 1,000 mussels that we could handle, maybe 240, 250. And that's a lot for us to be able to comfortably kind of handle that, manage that, and kind of move it through the system. And I think maybe our next step, is, and we've kind of done this a little bit in the other lab, is to begin to verticalize, the setup. So we're going to have multiple levels, like say 3 levels, 4 levels, and that would take up a lot less floor space for us. So, but I understand, the balancing the needs for big numbers versus say, focusing on some of the critically endangered species, like, like Tim had mentioned, they are focusing on some of these super critically endangered species, and they can be really challenging, to produce, just even a handful of animals. So I think each of the facilities has got different kind of goals, different things that they're operating under. So we are dealing as best as we can with the big numbers at both facilities. In the other facility, we're trying to do big numbers of, say, some of the actinomyia species. But, physically handling them and then balancing that, it is a challenge. And, we're just kind of, going, year by year. But I think probably for us, the next step is try to make the systems more vertical to take up less space to be able to handle more.
Need some hands from Harrison Lake. Whoever wants to jump in.
Yeah. I was just going to mention that the species that we do, the implicata here, we'll do them on herring, and it's a large infestation. We're getting tons of drop-offs every single day. So we'll estimate how many we have, and then we put them in a flow-through raceway with sand in it. And previously we just kind of left them in there until the end of the growing season, when they either were left a little longer till the following season, or they were dealt with dispersed into baskets at like 200 per, which, that's a long day. And so we're sort of trying to think of maybe more commercial-esque methods to handle some of these common species. That was a very hot topic at our, sort of just our Harrison Lake hatchery meeting that we had last week, just discussing how we might adjust our methods going forward.
Yeah, and I'll just give a little bit more on that. So for our implicata, we're getting anywhere from like 100 to 200,000 drop-offs. For all the other species that we do, our program is quite complex. But not everything is going out the door at tagging size. A lot of the projects that we're working on, we're giving incredibly small mussels in large numbers, just depending on whatever program it is or whatever grant or whatnot. So just because we're having like a total number of large number of drop-offs doesn't mean that that year we're handling that amount. And so it just takes a lot of planning and figuring out what our priorities are, depending on listings or which program it's falling under. So it's a lot of planning, not just for like where do just the initial drop-offs go, but what happens in 2 months, 6 months. And so it's just a lot of planning.
Anna, in that hatchery or in that raceway that's flow through, do you get a lot of deposition, organic deposition from
Yes.
the pond water?
Yes, and from the mussels themselves as well. Sorry, go ahead.
Are you cleaning that out? We tried to do a bit of that this year, or just had one batch where we had inoculated bass in a tank of flow through pond water, and took the bass out when they were done and just left them and wanted to see what would happen. And it worked for a while. We would sample the sand and a lot of small juveniles in there, but over time, it was just too much. With all the pond water going through it, it was too much organic deposition. And then it pretty much just choked them out. And it was at a scale that it would have been difficult, or we weren't, but it would have been difficult to try to keep it clean in any way. So we ended up losing those juveniles. So I was curious
Yeah.
as to if you had a maintenance for that.
There previously wasn't one. We're going to be doing things, I think, pretty differently with that species this season. Probably not stocking individual raceways as densely as we did before. And that's mainly because of the buildup. Especially towards the end of the raceway. These are big raceways. I believe they're 10 feet long, and I think they're probably 2.5 feet wide. So yeah, there's a lot of deposition in the back. When you stick your hand in there towards the end of the season, I mean, there was like, maybe you shouldn't say this, but I think there was like 3 inches of sludge on top of the sand. The ones that survived came up through that. But it went too long, so we want to be more proactive with that whole thing this season. Maybe try rearing some of those implicatas straight off the fish host in our ponds, see how that goes, since they do seem to be so hardy. We really don't touch them, or hadn't touched them once they went into that raceway. I pulled a few out periodically just to sort of try to see if they were growing at all. And I think we could have had even more success with the raceway had we used numerous and had we had more flow going through the raceway itself. Yeah. Possibly though, with our liming and fertilizing adjustments this season, it would be nice if we could push more food without pushing too much more water. Yeah.
Brian or Joe, y'all want to chime in?
Now, I was going to touch on one thing Anna mentioned, and I'll just be preaching to the choir about this, but Anna had mentioned about maybe not stocking those raceways as dense as we have. That's not always been the plan when we do that. Usually it becomes a matter of necessity because we don't have space elsewhere with the number of juveniles that we end up producing. So we end up having a target number, and then sometimes your target number is double what you thought it might be. So you're sitting with all these juveniles, and nobody's probably just going to dump them down the drain. So you try to figure out what to do with them. And if you don't have space elsewhere to go with them, you end up stocking at a denser number than you might like to. And what we've seen, at least in the raceway, and this gets into the decisions facilities have to make, is that sometimes you get almost no more survival if you throw 2 times as many individuals in there as if you just dump those other 50% down the drain, because there's a carrying capacity there that you're not going to really get beyond very much. And so that's where Jess gets into talking about going vertical and having more space. And then you start doing that, and you need more people to look at them. So, working with these, more fecund, higher producing species, Chris, that is a challenge for the facilities that are working with them, because it's always a kind of a game where you're chasing your tail in some aspects, and you become a victim of your own success because you have individuals from last year still at the facility while you're trying to get individuals from this year that you're producing into systems, and everything is kind of jamming up on top of each other until you can get them out the door. And so even with all the planning that you can do, sometimes you get one huge batch of animals that you do not anticipate getting based on your averages of production, and it throws you off immediately to start the season. We had that happen a number of years ago with green floaters specifically, where we had all of our boxes laid out for who was going where, and we got over double the number of green floater off of the 10 individuals we had compared to what we typically get. And we were sitting there with 250,000 instead of 100,000. And it became, what do we do with them? And then they jammed our systems up for the whole year. So that's always the trick with working with these animals, where you can produce, bigger numbers relatively easily. And we have that. Implicate is a huge producer. We can easily produce 300,000 off of one batch, no problem. But it becomes, where do you put that? When Anna mentions, you're getting into a commercial thinking with those types of things, some kind of huge natural system where they're out there, kind of where you have minimal eyes on them, but you can make sure that they're doing okay, so they just don't all crash on you as you're not looking at them for 2 weeks, and then everything just goes to hell. So that's kind of the trick with some of these, as opposed to the rare things where you're dealing with maybe 3,000 to 5,000 individuals for the year.
Yeah. Joe, did you have anything else to add since you're with the group, or is that?
Yeah.
Yeah.
Yep. So yeah, one of the things we do with our common species, because a lot of times we like to kind of start them off and say like our pulse flow box kind of system. But then we kind of have to, if we have way too many, or, way more than we were kind of thinking, we'll start to put them in the LSBs, which are a lot lower maintenance, and we can, they're a lot less complex. There's just, a bucket of water and an air stone. And you can, fit a lot of mussels in there. So sometimes that's an overflow. But we also learned some lessons this year with Eastern pond mussel. So we had a ton of that animal, and some of them went out in July, and some of them we kept in the systems just based on space outside in those later grow out systems, and it actually made a pretty big difference in terms of the ones in July were double the size. So sometimes, yeah, if you hold on to them too long, you're going to start to run into issues, especially when you're dealing with a common kind of hardy species. Yeah, we kind of have to start to change how we think about those.
Drew, you got something to say?
Yeah, I think so. Nothing probably of too much importance. It took me like 5 minutes to figure out how to raise my hand. But just to echo kind of what Jess said and some other folks have said, trying to delegate that, we ended up with a huge number of silicoidea this year, Lampsilla silicoidea. I think we ended up at some point with over 60,000 that are, 10 millimeters long at one point. And so we started shunning some of those animals to different places. We stocked some over in Virginia with Brian Watson, and we transferred some to West Virginia for their cage culture stuff. And we transferred some around to some other partners. and then put them out into baskets too. But we kind of ran out of basket space at some point with those. But I think that goes back to trying to find alternatives for those animals to where you're not holding on to them as long. I think shunning them earlier is a great idea if you can stock them out earlier. If you know that your back end survival is going to be pretty high. We were able to transfer some pretty small animals to some folks, some like 2 and 3 millimeter animals. And so, we've got a really large capacity built up on our boxes on the front end to handle those fluxes of large numbers of drop offs. And we kind of handle that by maybe not maintaining some of those boxes as thoroughly as we would, some of the others. And they do seem to do okay with that. So you can kind of I think get them up to where they can be transferred and then start transferring what you can. And then, if you incur a little higher mortality on those, you may have to just because you have so many.
Nice, thank you. Anybody else want to chime in on how they're handling these large batches of mussels?
Hey, this is Monte. I can't figure out how to raise my hand either, but so I'm gonna just talk. Just a couple of things I think just to think about. We're raising mussels now, at 10 to 50 times what they are in the wild, which 20 years ago we probably wouldn't thought that was possible. But so we do have lots of. I'm talking about the group as a whole. People are raising them very high levels. So I think something to think about is, what's the size goal that you really need? So like we can raise mussels in a small tray, and get them to 15, 20 millimeters, in 6 months, no problem. But that's at 50 per tray. That's still 150, 200 per square meter, maybe even higher than that. But we see just as good a survival at keeping 1,000 in that tray. They're just smaller. But they still, after the second year, they get to the size where they're 15 to 20 or even bigger, 20 millimeters. So you have to kind of weigh the cost and say, well, do I really need them to be 40 millimeters in 6 months? Or can I just get them to the size where the survival is very high and it's more cost effective to have, 1 or 2 or 3,000 in a small tank versus 100. But some of the mussels that people are dealing with, like, some ones we're dealing with, there's not 100 in the wild. So sometimes it's more cost effective to keep them at really low numbers so you can get a larger size quicker for a small number, because that's all you're dealing with. For some animals that I work with, I might not even have 500 larvae to work with. So I'm going to keep those at a lot lower density than I would something that I have lots of common animals. I want to take my losses on there. I'd rather have, survival very high and them being a little smaller, but still survivable size. And so you have to kind of weigh the cost with your space. We can handle about 360,000 at a time, but not at 50 per tray. 50 per tray, we're going to be down to just 4,000 or 5,000 mussels. And so, we just have to say, well, it's not worth it to have that small, that huge, if we can only handle 5,000 mussels. Then we'd be only working with 1 or 2 species. So you just have to kind of weigh the cost. And I think Jess hit on something as well, that, what's our ultimate goal? How many mussels do we really need to put out into the wild to establish a, survivable recruiting population, and then we can leave them alone in the wild. And so I think we all need to answer those questions and look and see, what is the ultimate goal? If 50,000 for an animal is all we need to raise, and that's going to 100 times what's in the wild, that may be an adequate goal. We may not need to raise 5 million of a species. Maybe that is the goal for like the Chesapeake Bay and Leptio complanata. Maybe that for the engineering aspects, you maybe need 5 million mussels out there over a 500-mile radius of streams. So I think you just have to come up with your goals, and then you have to look at your space and say, okay, well, I will take my losses if I can get higher numbers and they're half the size. I'll be fine with that because the survival is going to be the same. So that's just my thought on maybe a way to look at it. One thing that we do look at is I do a lot of monitoring, and I'm looking at what densities that I see in the wild where I see recruitment for multiple species. And so I'm tracking that over now 20 something years. And what I'm seeing is I look at different species that are closely related and say, okay, if I have the best model out there, how many individuals do I need to establish, let's say 40 riffles in the Green River, to be able to establish what I'm seeing at the best spots? And then I can come up with a kind of a number to pick. And then that's going to be my goal over a 10-year period. And so then you can spread your recruitment out, and your brood stock is every year. You stock them at stockable sizes like they would be in the wild. And you can come up with a game plan that works with your facility. And so you don't have to do them all in 1 year. Sometimes it's better, I think, to spread them out over several years. And then you put, you're stocking animals at a natural type of stocking density that would normally recruit. We don't even know how many recruits are out there in most river systems. So how can we pattern how many we're putting out if we don't even know how many are there in the first place? So I think you have to really look and see how many recruits are out there. Okay, what's the best model we got for that system for this species, and come up with a game plan and do it over a 10 or 15 year period. And then that gives us some goals, and then figure out how to reach those goals and still be able to work with some of these really rare ones. And it just takes a lot of planning. And I think we're all at a really good stage where, oh gosh, 15, 20 years ago, we couldn't do anything. We didn't have these problems of figuring out, okay, we got too many. We don't have enough. We can't even do it. So, and with some species, we're still that. So I think there's just a lot of questions that we all need to answer, that we need answers for. And I think every species is a little bit different. And I think the next phase of mussel propagation is going to be like establishing a national mussel system. So we get 4, 5, 6 hatcheries doing the same species over 4 or 5 years, and release those all over their range, and then all of a sudden, oh gosh, we're done with that species. We can go to another species, and some of these animals, there's only one place they're found, and there's 50 animals left in the wild, and so that's a different system, a different way to look at it. But that's just my thoughts on some of the differences on how you manage your hatchery and kind of what goals are and how do you address those.
Thanks, Bonnie. Anybody else want to weigh in on that?
I'm going to add a little bit to what Monty was saying, just real quick. Like the clinch, we've been surveying the clinch and the POW for, decades. And, there's maybe 6 species, a few of us in the call can just name them off the top of our heads, that are at higher densities, meaning they're like, 2, 3, 4, 5 per meter squared or more. And I'm sure Monty and, others have similar data, but then 80% of the species that occur in the clinch are probably occurring at densities of less than 1 per meter squared, many of them below 0.5 per meter squared. And here's the thing, they're scattered out throughout the river. They occur on the sites, the different, high quality sites. They've been there for decades. They recruit. We occasionally see the recruits. And just, using that kind of knowledge to come up with, some targets like Monty was suggesting. I think that, we might be surprised that for some of these species, really, they're just not going to maintain themselves at 2, 3, 4, 5 per meter squared naturally. They're just not going to do that. There's some limiting factor out there. Fish host, or the way they interact with their fish host, their fecundity, whatever it is, there's this limiting thing. And they're just not going to be this super abundant species. So we need to be realistic about that. It's like we need to take that, that data from the field and be realistic about what the targets are and what we're shooting for, and then translate that into, hatchery operations like Monty was talking about. And I think we've got the data to do it. We just need to kind of sit down and kind of think it through for, different rivers. And, and like Monty said, there's some species like elliptial campanata and others, for different reasons, we want bigger numbers. But for many of the species that we're working with, the numbers might be a little bit lower than we might think. So,
thanks, Jess. Well, did anybody else have any other questions on their mind? Either questions for any of the presenters or questions for issues you've got at your own hatchery? that you want to raise. All right. We got one. Isabel.
Yeah, so I wanted to ask the labs that use black cow or worm castings, um, are you guys microwaving or boiling that before you're adding it to your containers? And has anybody tried not boiling or microwaving if that's the only way we're doing it?
Hey, so we microwave ours. And when I first started, I think was Rachel's first season using it. And she was freezing it without microwaving and then using it in the systems. And we had crazy outbreaks of all sorts of weird stuff, like worms and fungus and just all kinds of really wild things showing up alive in our systems that were not mussels. And some of that might have been that we were still on the old intake water, so we were getting a lot of weird stuff coming in our water too. But when we stopped using the compost, we stopped for a while and then kind of rethought how we were doing it and started microwaving. But when we stopped the compost, a lot of that did improve. So that's the only time we've tried without microwaving, and it was just being frozen.
And thanks, Sierra. And like I mentioned, we have gone to an Instapot, and that is great in that it contains, so you don't have this boil over of mud all over your microwave. I'm trying to contain that. So so that's been great for us. We've got a couple of those if we're really cycling through a lot of sediment. But yeah, I would recommend trying to sterilize it somehow.
This is Monte. I was just going to weigh in on that as well. I really think trying to standardize your foods as much as possible is very helpful. And one way is to sterilize that material, and then it's very consistently applied as a food source. And so I think anything we're doing to kind of repeat things year after year, the more we can standardize things and keep problems from happening, makes it a lot easier. So that's one of the reasons we sterilize it, because I tested it without sterilizing and with, and I didn't see really any difference. I didn't really have any problems with any worms or fungus of the unsterilized stuff. But I think, it depends on how long you're keeping that stuff in a bag. It could go bad after a period of time. So, we try to use ours up over a period of a year and then get a new bag, and just be consistent about using it. I really, if you ever look inside of a mussel stomach, you see a lot of that organic material. That's when I started using it, because that's what I saw in the stomachs of mussels. And so I wanted to replicate that in a standard way. And, we used to get sediment from streams and ponds, and we couldn't be consistent about that material. And so, worm casting, organic material, whatever it is, topsoil, it's just consistent source of this organic material that a lot of mussels need. Some of them I don't think probably need it as much, but a lot of them do. And I've seen a tremendous difference. But again, just, sterilizing it is the easiest way. I'm really curious, Chris, about not using the microwave. So I'm curious about your other way of doing it. That might be actually a better way than microwaving it.
Yeah, just for the mess, it certainly was. But yeah, and you can do a bigger batch at a time. In an Instapot, it can hold however many liters of sediment at a time. So and you get all the pressure, and it's essentially an autoclave. It just happens to be cheap and affordable and available at your local Walmart. But thanks. Yeah, great question. Any other questions? Any other issues at your hatcheries you want to bring up to the group here?
Hey, this is Tim. I just wanted to quickly weigh in on the bigger conversation before, and we're probably the most space limited lab as far as big batches of anything goes. We could take up all of our space quickly, and that's happened in the past. And we've talked about quality over quantity in a lot of cases seems to be important. And when you think of drop off period like in a bell curve, one thing that we've done is you take a lot of the earlier drop offs. Yeah, they're moving around, but we're not in the peak yet. Kind of put them to the side. And then when you're really in the peak of it, that's what you want to really give your prime space and, your prime real estate in the lab to. And then maybe like after 2 or 3 days of that prime drop off, then the rest of it kind of goes back to, you guys are the late ones, and so you're not going to get the prime space, and at least that's worked for us in some cases. It's worked out where we end up with those peak drop offs, and containers just seem to outperform the others. And another thing is we have all those green tanks I showed around our pond. In cases where like we produce too many epioblasmas or something that we weren't planning on, similar to how Harrison Lake was describing happening with the green floaters. When we max out the space that we had planned to give them, we'll just start taking them directly out and putting them in sand in the bottom of those tanks with pond water. And it is. We talk about inside, we've got elementary school, middle school, and then they graduate outside to high school. Well, you can think of this as they just go straight to one of these religious schools where, K through 12. And sometimes it works out and sometimes it doesn't. And, you'll end up with a big batch of really good ones sometimes, and other times you get zero. But they didn't take up space and cause you to have more mortality in your prime space inside the lab. And so, we have cases of where we've only ever let 3 things go, like Philoderma intermedia. We've let 3 go, and all of them look beautiful when they went out. And we found 100% surveying for them. We found them alive and well now 2 or even 3 seasons in a row, all 3 of them. And then there's things that we put out in huge numbers, sometimes a cohort of 20,000 or something, and 3 and 4 years later, we're not even finding them anymore. So quality over quantity might be a really important thing to think about. You want animals that are going out there healthy, and they're going to perform and be normal out on the shoal. Like just numbers and demographic models and things, they do matter and they should help guide us, but they're not the end all be all. I think the most important thing is putting out quality animals that are going to survive and reproduce on their own. That's really the ultimate goal, is they take the reins and they're going to reproduce, and we don't have to keep on stocking on top of them anymore. So that's my 2 cents.
Nice. Brian, you want to add something?
No, I had a kind of a specific question for all the various facilities, and some people touched on this in their presentations, but with the systems you're using where you're putting newly transformed juveniles into sediment systems, whether those be pulse flows or buckets or whatnot, what size sediment are you all using to start off with, and what temperature are you raising those juveniles at typically? I'd just be curious around the board what everybody's doing on that.
This is Monte. I can start with, we use a sub 100 micron sand as our main substrate, and that's because all the mussels are larger than that, and it's much easier to separate the mussels from the sand and the substrate. And so you add in the food into that mixture, and that's what provides the basic source of the food. And I really think that small sand is something that's missing in a lot of streams today. And I'm talking about really small, less than 100 microns. And so that's just perfect to catch all the particles in between the interstitial spaces. And but many people have raised stuff without any substrate at all. So so I think there's some species can deal with it a lot differently, but we use very small sand.
What's your temperature? What's your temperature usually at Monte?
In the spring, we'll start them at 22 to 24 degrees, but ideal is 24 to 25 degrees is where we try to keep them. But sometimes our greenhouse will get a little cool at night, and so they may get down to 22 or 23 at night, and then warm up to back to 24 to 25 during the day. That's our typical temperature range. And these are going to be for brand new juveniles till they're about 3 to 4 millimeters. And so when they go to our branch hatcheries, you know, the temperature range is anywhere from 25 to 30 degrees centigrade, depending on time of year. So we don't control that because it's outside, you know, lake environment. And but in our controlled environment for the first few months, you know, we want to keep it not too awful warm. You know, it might in the middle of July, it might get 27 degrees in the tank one day, but it'll cool back down at nighttime to 25.
I'd also ask you to add what type of sediment y'all are using too. I think I've heard quite a few different ones while we were talking about this today.
To answer Brian's question, we are like Monte, we're using less than 100 microns sediment that our juveniles start out in. And again, that's it's about three quarters worm castings and 1/4 limestone, ground up limestone, and that's all less than 100. We're starting our juveniles off. Well, starting them off at the same temperature that they come off the fish, and that might be starting at 22 and then raising them up to 24, 25-ish. And I think I remember others saying about the same thing, about less than 100 microns was pretty common, I think, across facilities, Brian. All right, any other questions for the group? Issues at your hatchery you're trying to solve? There's a note in the chat from Minnesota DNR. They're using starting everything in less than 150 micron of locally sourced mud and transitioning to black cow. They're transitioning to black cow to start out. Kind of the same thing. Starting them off at temperatures where they're coming off the fish, near room temp, and slowly raising them up to 24-ish. All right. I guess I just had one other question that I wanted to pose to the group, and I was kind of alluded to it when Monte was talking earlier. I'm curious how other hatcheries are managing moving mussels when it comes to the end of the growing season, and you're dealing with animals that may or may not be ready to go out into whether it's wild water, winter temperatures, what have you. What temperatures are you comfortable sending mussels out into into dealing with the winter temperatures? And what do you do if they're not quite there? We have some that we're rearing inside, and now we got to the end of our growing season and we had a bunch that were around 3 millimeters, the ones that we did later in the year. And I didn't want to send them out to, go through winter, but now we're raising them up and at room temperature, we're feeding them, they're growing, and now they're starting to outgrow our indoor space because they're getting cramped in our indoor space, but I can't really, they're at 8 or 10 millimeters now, and I don't want to can I just send them outside and, with the water out when our flow through water is at 5 degrees C? Trying to figure out how to handle all those animals. I'm just curious if anybody had overall strategies on how they dealt with things like that.
So Chris, this is what we do at AWCC. We have a big tagging party normally every September. And we get as much volunteer help as possible. As we're going through every species, there's always a portion that are too small to tag. Almost every species, across the board. And so we set them to the side at that time. A lot of times we take them right back out where they were again, where the tagged ones will go to a place where they can begin to purge out a lot of their guts and stuff before we go and stock them. But they're tagged and bagged and ready to go, where the others will set maybe back there for another week or two. And then we have a system which we didn't share with the group, but it's like the one we're holding the brood stock in, and we don't feed it. It's just fed the pond water, whatever temperature the pond is at, then it's at the mercy of the pond. And through the whole winter, they experience true winter and hibernating inside because we're not continuing to try to grow those animals. We just say you were a little bit small, and so it's gonna be like you went out in nature this small, you're just gonna have to deal with winter. And we just we do not feed them the nano and shellfish again. Because when we've tried that in the past, when they go to a complete wild diet out on the pond and then come back in and you put them on shellfish and nano, we had high mortality from doing that. It seems like whether it's their gut microbiome or whatever reason, they have shifted to wanting to eat a wild, they have an appetite for wild algae and things at that point. And so anyway, we have really high survival just letting them be at the temperature of the pond water in that system. And then they go straight back out to the warm pond next May. And so, yeah, there's space issues with that, but the survival is really good in that situation. And then any animals that were never big enough to take out to the pond in the first place by cooler times of the year, they just have to remain inside getting fed nano and shellfish all winter long. And a lot of times it's not ideal. Like they do run into a wall, like what you're experiencing. Like we definitely experienced that. And that's why we had created that kind of transitional trough or planter box system, was to kind of give some more space to animals in that situation. So we're still learning with the little space we have what works and what doesn't. But I can't be more adamant that, like, if you guys have put things on a wild diet at all and they're too small to stock, don't bring them back in and put them back in a recirculating system with nano and shellfish only. If you have a live algae bioreactor the way Monte and some of the other labs do, then that's probably different. They can probably do just fine in that situation. But I heard Monte say like he left any animals that were too small for another season in his floating cages. Well, it's the same idea, basically, what we're doing. We're just keeping them. They would have experienced winter, even if they were little, they would have experienced winter in the wild that way. So they should be just fine and adapted for that. That you don't have to keep growing them just because they're with you.
So Tim, what size are you comfortable putting them in that system where they're just going through winter?
They're normally Bissell thread or bigger. We tried to put them outside, obviously, at that point and transition them onto that diet. So they're not microscopic anymore. Anything microscopic or up to like that 1 and 2 millimeters, we would have never put them out in that situation yet. They would have still been in the sediment tanks or those pans.
Chris, this is Monte. We've actually tried releasing mussels at in our wild tanks at 1 millimeter, 2 millimeters, 3 millimeters, and anything under 3 millimeters, we didn't have very good survival through the winter. So there is a size that is optimum to get them through the winter, and I really think it's more like 5 to 6 millimeters. If you had a bunch of 4 millimeter animals, I would try them and keep them in the wintertime conditions. And like I said, I don't think it hurts you to keep those animals cold, and they're just not going to grow as much. They'll do a lot better if you just keep them all the same size and let them stay cold and let them warm up until you can release them into the wild where they actually have a food supply. And so you can actually track their, your temperature, and you actually warm them up as the water is warming up outside. And then so that way your transition to the wild is very, very similar to the same temperature, because I think you can have some temperature shock stress that is associated with moving animals. And I think if you try to put them in too late in the year, let's say November, it's too cold for them to get acclimated, and they're using all their energy trying to burrow up and stuff and try to switch diets, and it's too much for them. So we kind of use the policy of, we want to put ours out while there's still a little bit of growing season left. Even if they're just a little bit smaller, they're going to be a lot better off. But if they're below that threshold size that we need to tag, we're just going to keep them for a whole nother year. And it's just much easier, and it doesn't really cost us that much more to do that, depending on what species we're dealing with. But that is a common problem, I think, that people are facing. What's the best time of year to release mussels? What's the minimum size that you try to get to release, and what's the minimum size that you can keep them? We don't keep anything at our facility in Frankfurt during the wintertime. We used to do that. But it's a lot of extra work, and we didn't really see that much of a benefit. So we just move them to our facilities, let them experience the cold winter. Sometimes you'll get a little period of warm in December and January where they may grow just a little bit, maybe not much, and then all of a sudden it gets cold again. So that's the normal temperature regimes that they're used to. And you'll get a little algae bloom in our lakes and something, in the middle of the winter time, just a day or two. And that's just enough food to give them a little bit of nutrition during the wintertime. They're still feeding and pretty active in the wintertime, surprisingly.
Michael.
Yeah, at least the way we handle it in Alabama is, it's about the middle of September. We kind of judge that's enough time to get them transitioned and get a little growth in before the cold weather hits. So we really move everything out, regardless of how big it is. Like, we have some pluribemas out in the pond right now that the average size is probably 2 millimeters. They're on a 500 micron mesh, and they're still alive. The pond's 5 or 6, like 6 degrees today, but they're hanging on. And I assume when the water gets up about 18 or 19 degrees in the spring, they're gonna pick it up again. But we don't have the option of keeping them over the winter without stuff freezing and having to maintain stuff. So we just sort of just push them on out and wish them the best. And sometimes it works, sometimes it doesn't. But more often than not, they're pretty hardy.
Hey, Jackie.
Hey, so depending on what species it is and like how much we're actually dealing with, we have a couple different things that we do. We try as best as we can to get them out at the appropriate sizes, whether that's into our shad house through like our U pans, or if they're going into baskets. So we try to get some of them out quickly, so that the whole batch is not being held over winter, whatnot. But we do hold a couple species back only because of the timing of when they're spawning. So Campanata, and James spiny mussel, that's like in June and July. So they're not as far as long, obviously, as some of our other species that we do, March and April. So we can push them, but they're still not big enough to go to the next system. So we do overwinter in our tub wellers, our upweller systems. But what we do is we don't keep them at that higher temperature we normally would. We do bring the temperatures down over winter, because otherwise we could grow them really big, but if it's the middle of the winter, then you're back in the same spot of like, I don't have any place to put them because they're too cold if I put them out into our flow through system or into baskets. So we try to like mimic some, not like cold temperatures, but we do bring the temperature down a few degrees over winter.
Thank you. Does anybody else want to take a crack at that one, or have any other questions that you want to ask?
I'm going to mention one more thing about that.
Okay.
Something you can do that we've done before is you can warm your tanks up in the fall for those ones that are real small, and keep them an extra month or so and get them to that, a little bit larger size. And then over a period of a couple of weeks, slowly cool them down to your outside temperatures and then move them outside. So that we've done that before because, we wanted to move them in September, early October, but they weren't big enough. So we kind of held them a little bit longer than we normally would. And then we cooled them down over a period of a week or so to match the temperatures that are outside at our other facilities and then move them. One thing I will say that I didn't really hear anybody talk about is one thing we never do is we never move them in the middle of the summer when it's the hottest times of the year. I feel like that's the most difficult time to transport mussels when the water temperatures and the air temperatures are so different. And just dealing with handling the animals outside in the heat sometimes can really cause some problems. So kind of waiting till September is also got a benefit of the air is cooler, the water's a little cooler, and you don't expose the mussels to a stressful time of year like the middle of the summer.
Yeah, thanks, Monty. That was, I'm glad you said that because that was something that I was considering. Like, is that a possibility? So these mussels now that I've got in this system, in my second system that are reaching about 8 or 10 millimeters, I'm continuing to feed them. And the bigger they get, the more I feed them, the more crowded they get, the more stunted they get. But now that it's January, can I start to cool them down by slowly bringing in outside water, winter temperature water? Can I slowly cool those down here in January just to save 2 months of food because they're eating a lot of food and getting a lot more crowded. So I'm just curious, is that something, Monty, you think I would be able to do? Or do you think I should just keep feeding them until April when they go, out and get our full flow through?
Yeah, I mean that's definitely a good plan because there's a high correlation between temperature and how much they're growing. So you just have to reach, what's the low temperature that you want to, reach and get to that's easy for your hatchery to maintain. And I really think the temperature cutoff is anywhere about below 15 degrees C. So I think once you get between 10 and 15 degrees C, the growth is so similar that there's not much difference. So just whatever is the easiest to handle for your lab, you don't need to go to 6 or 7 degrees. You can go down to 10 or 12 is fine. But once you start getting above 15 degrees C, and again, depending on the species, some species that live in that really colder water, they'll start growing at 15 or 16 degrees a little bit. But ones that live in typically warmer water streams, it's going to be more like 18 or 19 degrees before you see really any significant growth. So you really kind of have to know your species and which ones you're dealing with before you can address that. So you have to look at the species and figure out what is my target low temperature that I can get to with that, and how much food will that save me. So you have to look at how much food is in the tanks, how much are they removing daily, and then kind of look at that, what's your balance and what works the best depending on how much food you're raising.
Well, I guess that's it. And we will wrap up. I'm curious if you have any feedback that you want to send to me on how this went, on maybe something similar that we might do another time in the future with a similar group. I know Jackie suggested over email that maybe we could do something like this with an in vitro group where we're talking nuts and bolts. So if you have any feedback along those lines, I'd love to hear it. Just email me. But with that, I will again thank everyone for participating. Thank everyone for attending, for putting your time into your presentations. I wish you all the best in getting ready for this upcoming busy season, and hope to see you all at FMCS. Thanks again.